Good morning, everyone. Welcome back to the second day of the workshop, National Online Workshop on Pharmacovigilance. Hope you had a very good sessions yesterday and you enjoyed all the sessions and got some insights into the pharmacovigilance, especially on how to report these adverse drug reactions in the VG flow. And followed by, we had the sessions on signal detection. So today we are going to have some more interesting sessions, especially from industry perspective. You can understand how exactly the pharmacovigilance is followed in industry. And under that, in the first session, we are going to have how to go for the data mining in some of the pharmacovigilance databases which are available freely in the platform. And we can say them public drug safety databases. So we have here with us the first speaker of the day, Dr. Subhish K. Vishwam. And currently he is working as a senior associate for scientific writing in Digin, Bangalore. And previous to that, he was working as an assistant professor at MS Ramaiah University of Applied Sciences, Bangalore. And if you see his experience, he is an experienced academician and researcher as well, with a demonstrated history of working in the hospital and the healthcare industry. He is skilled in drug safety, signal detection, data mining, clinical research, pharmacovigilance, dose calculations, identifying and rectifying drug-related problems in the clinical trials. And also he's having a very strong academic background with his Doctor of Philosophy from MS Ramaiah University of Applied Sciences, which is mainly focused on the pharmacovigilance in psychiatry. So I can see his publications, especially in this area, in the pharmacovigilance and data mining in various drug safety databases. And I hear that many people are using those articles as references while they're conducting research in the area of pharmacovigilance. So with this brief information, now it's my pleasure to welcome Dr. Subhish for delivering the lecture on this topic, data mining in public drug safety databases. Now the screen is yours, Dr. Subhish.
Yeah, thank you so much, Krishna. It's a privilege to be, a speaker in this conference. Webinar. There's a small correction. Actually, my terms with Indigene officially end last week. I'm not associated with Indigene now. So I'm taking a small break, and soon I'll be joining in a new institute. So I just want to correct my designation. Yeah. Let me know if my screen is visible.
Yes, Subhish, your screen is visible. You can go ahead.
So starting with the mandatory disclaimer, whatever I express here is my personal view and no relation with the designation I hold, neither the organization I work for, and it is purely educational purpose. Okay, let me start with a small case. A 65-year-old male patient admitted in a hospital with chief complaints of decreased urine output associated symptom, and he's a non-case of type 2 diabetes mellitus and hypertension. He's on enalapril, canagliflozin, and amlodipine. So it shows that he's having an acute kidney injury. So which drug you will be suspecting here? Usually when we see AC inhibitor or in this particular class of drugs, we know which one to suspect, isn't it? Let's move to the second case where a 60-year-old male patient on case of T2DM, dyslipidemia, hypertension, diagnosed with rhabdo. So the patient is on linagliptin, insulin, atorvastatin, amlodipine, and enalapril. So what do you think? Which drug? We'll come back to the answer. I'm just going through the case, and we'll come back to the case after this. Third case, a patient came with the complaints of dark stool, abdominal pain. So he was on aspirin and clopidogrel, Sinalset, metoprolol, insulin. So on examination, physician confirmed that he's having a GI bleeding. So which drug? So usually, when we speak about kidney injury and AC is there, ACE inhibitor is there, we usually suspect enalapril. So my question is, why you're not suspecting canagliflozin, or why not amlodipine? When we speak about rhabdomyolysis, we always say it is because of statin. Why not other like insulin or amlodipine or enalapril? Then when we say GI bleeding, we always suspect aspirin or clopidogrel. So why can't we say that it is because of Sinalset or metoprolol or insulin? Yes, we need a causality assessment, but usually in usual pattern, we always go with the expected thing, right? We know we have a priori information and we will always go with that. So do you think it is a correct practice? For me, it is not a correct practice, and because of this, we need a little advanced technique to identify. Let's say the first case, canagliflozin actually can cause kidney injury, but it very recently came to the picture. So if everyone is reporting for enalapril or whichever drug is co-administered with canagliflozin, how we will identify whether canagliflozin is causing this or not? Okay, so that will be the main focus of this particular talk, not about canagliflozin. How to identify a signal? How safe is a drug? So if a drug got market approval, does that mean the drug is safe? Definitely not. Correct. That's the reason we have post-marketing surveillance. So the adverse drug reaction of a drug will be same throughout the course of its life. No, correct. The adverse drug reaction keep adding to its profile. So that's the reason how important is post-marketing surveillance. So moving ahead, though the benefit risk profile of a drug is assessed during the clinical trial, a newly developed drug adverse event cannot be predicted completely. So what I meant by this is, let's say an adverse event which have a frequency of or incident rate of 1 in 10 lakh. So you must be knowing during clinical trial maximum how much patient will be exposed to the drug or how much volunteers will be exposed to the drug. Hardly 10,000. Correct. So what is the probability that we will identify this reaction? Very less, isn't it? So how to identify this reaction which having very less incident rate but it is life threatening? Okay, yeah. So the benefit risk profile of a drug is completely known only during the post marketing, and it will keep adding to it. And post marketing surveillance will reveal unidentified or unreported adverse event of the drug. Yeah. So what is signal detection? So I know yesterday Dr. Vaidya was explaining everything all this, but I'm just I'll just rush through the slides and we'll have a small discussion. So whoever missed yesterday's session, they can use this particular session also. I'll try to make it as simple as possible. Yeah. So what is a signal? So according to WHO, signal is defined as reported information on possible causal relationship between an adverse event of a drug, adverse event and a drug, of which I made it red, so that is very important. The relationship is unknown or incompletely documented previously. So if you're coming up saying that, say paracetamol can cause liver injury, that's not a signal because that information is already known. So if you're saying that paracetamol incidence of causing liver injury is significantly reduced, so that is an incomplete or a new information where we'll consider it as a signal. Yeah. So what is data mining? So safety signals are usually generated through data mining algorithms, and this is nothing but a knowledge discovery in a database. So data mining is a systematic examination of reported adverse event, and signals are usually generated as a usually generated as a statistical score. So usually we'll say a signal strength of 4, signal strength of 6. We will be explaining it as or expressing it as a score. So those score indicate the strength of association. So when someone says that the signal score is 4, that means that compared to the other drug, this particular drug is 4 times more associated with the event. Okay. Yeah. So the use of data mining technique can broadly group into 2 main areas: identification of a new effect of a drug and appropriateness in the drug use. Oh, let's see. See, let's assume this is a database or a collection of information. Let's say it's a data. So previously you were not understanding that there is a sequence which is keep repeating, correct? If you see this sequence, correct, it is keep repeating. But if you look like this, it is not possible to identify, correct? Even my name also repeating. So this is something which we are going to identify, data mining. We are mining the data to identify a particular sequence or particular relation between two. Going ahead, why signal detection? So identification of a rare adverse event which may fail to identify in other conventional way. So as I told you, like if you want to identify a very rare event which having an incident rate of 1 in 1 lakh or 1 in 10 lakh or maybe 1 in 1 crore, it is not possible to identify. Just assume you are going to hospital and screening 1 lakh patients or 1 crore patient. It is not possible, correct? I'm not saying it is completely impossible. It is practically difficult. So in those conditions, this disproportionality analysis will be come to the picture. So also to identify complex relationship like association of asthma and Johnstone syndrome, serum sickness like reaction for cefaclor, T-cell non-Hodgkin lymphoma reported for affairs with respect to tumor necrosis, TNF-alpha inhibitor, then drug-induced gingival overgrowth. So these reactions are, it's not very common or very commonly used drug. So these kind of things can be easily identified using a database. So what is disproportionality analysis? Disproportionality analysis is nothing but whether we are identifying whether a particular event happened by chance or by coincidence. Okay, or we also trying to identify the particular signal what we generated. Is it because of the drug, or is it because of the comorbid condition, or is it because of the co-administered drug? So this is a very famous example about signal. What do you mean by a signal, or what do you mean by disproportionality? Okay, so let's say two tiger in a zoo. When we say any information, any statement, what our mind will do, it will try to picturize it, correct? And you have, a priori information about it, correct? Must have visited a zoo, and you know that two tiger in a zoo is quite expected, correct? But how about two tiger on a road? Until otherwise you are, crossing Bandipur or any of the wildlife sanctuary, you are not expecting two tigers on a road, isn't it? So this is something disproportional, correct? Let's take another example. 5 girls in a classroom. Yes, you can picturize it, but 5 girls in a boys-only classroom. That is not what we are expecting, correct? So you're always comparing a prior information. You have an expected count, correct? To that expected count, what you're trying to do, you are trying to correlate with the observation. Okay, so let's take an example. You have 5 reports of Y reaction for X drug. So my question is this 5 is a greater number? So how will you tell this? Is it a greater number? If you say depends, that's what the answer I'm expecting here. What depends? It depends on how many people get the adverse event in general, and is everything common, or is this particular event is common or rare? How many people take this drug, and even though they take the drug, how many of them will get the ADR, and in that how many of them will be reporting? You see, it is practically very difficult to get these numbers, correct? So what we can do? The idea behind disproportionality is to use the database of spontaneous reports to compute the number you would expect if a drug and event are co-administered by chance. Okay, so once we have an expectation, we can evaluate the observation in light of expectation. Please don't get confused. It's very simple. What I'm trying to say is, from a database, we will try to identify what is the expectation of a drug with respect to that particular event. Then you have an observation. You will compare this observation with the expectation. And when there is a disproportionality, that means out of proportion, then we will call it as a signal. Yeah. So I hope Dr. Krishna must have explained you this very clearly. I'm just briefing it. 2 into 2 table, correct? So when we speak about this, A, B, C, D. What is A? A is the number of reports containing both suspected drug and suspected event, where B is the drug of interest, but because of the other drug, and C is the event of interest because of the other drug, and D is the adverse drug reactions of all other drug and of the all other event. Okay. An example, very simple example. Let's assume, we have 45 people in a room. 10 of them ate cake. Okay. In that, 2 of them got regurgitation. And another 7 who didn't eat cake also got regurgitation. Let's make this 2 into 2 table. Okay. Yeah. So how much will be the count here? Correct. 2 of them who ate cake got regurgitation, and total 45. Then a total of 10 of them ate cake. Then so here it will be 8. Correct. Yeah. Then 7 of them who didn't eat cake also got regurgitation. 9. It will be 36. So this will be 35. Correct. So minus 7, it will be 28. Yeah. So this is how we will make the table. Okay. Yeah. So let's take another example, and I'll just brief you what you mean by reporting odds ratio, or proportional reporting ratio, or relative reporting ratio. Okay, when we say, I always say that observation and expectation. Correct. What do you mean by that? How will you identify that? Let's take this particular example. This is actually a real example. So ciprofloxacin and epidermolysis bullosa. Okay, so this is a skin disorder. We are suspecting, this is a live case we got, live incident which happened in our database, and we are trying to identify whether it is a signal or not. Okay, so ciprofloxacin associated with epidermolysis bullosa, we had 3 reactions. Okay, and there are total of 26 reactions of ciprofloxacin in the database, in our database. Okay, this is not FAIR's database or Vigilize. This is in the hospital database. Yeah. So we have 6 cases of EB, and this is the 2 into 2 table. So what is the reporting rate of EB? So we have 9 reactions of epidermolysis bullosa out of 1000. So what will be the reporting rate? 9 divided by 1000, that is 0.9%. What do you mean by 0.9%? Which means that if you take 100 cases, in that 0.9 cases will be EB. Correct. So what will be the expected rate of EB associated with ciprofloxacin? Since ciprofloxacin is in the same database, so this 26, correct, there are 26 cases of ciprofloxacin. In that 26 case, 0.9% will be EB. Correct. So 26.9% supposed to be EB. So what is the EB associated with ciprofloxacin? What is our expectation? Our expectation is, in that 26 case, 0.23 will be EB. But how many EB cases we got? We got 3 cases. So what we will do? We will have observed divided by expected. That means 3 divided by 0.2. We have 12.8. So this is the signal strength of ciprofloxacin associated with epidermolysis bullosa. Okay, so what is the significance of this 12? So it means that ciprofloxacin and epidermolysis bullosa are 12 times more associated when compared to the other drugs in the database. I hope it is clear. Okay. You don't have to go ahead and do all these calculations. You can directly use this particular equation. Next is PRR, proportional reporting ratio. So in case of PRR, what we'll do is, see, we are not sure about the drug of interest. So when we calculate PRR, what we should do is, we will usually avoid, these numbers. That means the drug of interest number in the calculation. Okay. So to calculate the expected rate, instead of using 9, we will use 6. Why 6? We have total 9 cases. In that 9 cases, 3 we are uncertain. 3 we are thinking that it is because of, we are expecting. Okay, we are not sure. We are thinking that it is because of ciprofloxacin. So we are not using these 3. The remaining 6 we will be using. The same way, ciprofloxacin caused 23 other adverse events. That also we won't consider. So how we will calculate the reporting rate of EB? 6 divided by 968. Which says that the reporting rate is 0.61. Okay, yeah. So the expected rate of EB of ciprofloxacin, we have 26 case. So 26 case into 0.9. So our expectation is 0.16. Observation is 3. So what is our signal strength? Sorry, this is not RR. This is PRR. Okay, this is a mistake. Yeah. So our signal strength is 18.62. Okay. So which means that it has more, signal strength. It is more associated. Next is ROR. ROR, same way how we are calculating the odds ratio, same equation. Yeah. So what do you mean by this disproportionality, or A, B, C, D? this is the drug used in the population. Okay. Yeah. And in that, this many people got ADR. And in that, this many ADR are being reported to the database. Okay. So why I'm putting this particular slide, because to make you understand that the main drawback of this particular study or this particular research, we have a huge, this many people reported or this many people took drug, but what information we have is only this much or maybe less than that. Okay, so please keep this in mind when you are doing this research. Oh yeah, another algorithm is IC. This is difficult to calculate but more reliable data mining algorithm, not algorithm, measure. Yeah, so few of you must not have understand how to calculate it. Doesn't matter. This is a computer era where you don't have to sit and calculate all those things. You just enter the data. The computer will calculate and give you the signal strength. But what you should know is how to interpret it. Okay, so there is a threshold. Whenever the ROR minus 1.96 SE is standard error, if that value is more than 1, we will say it as a possible signal. Okay, when PRR value is more than 2 and chi-square value is more than 4 and minimum of 3 cases are being reported, then we will calculate or we consider it as a possible signal. And any positive value for IC, that also will be considered as a signal, not signal, a possible signal. Okay, so this is the only thing you should remember. You try to understand the concept, but if it is very difficult for you to understand the concept, what you can do is you try to remember the threshold of this. So what was the PRR value of ciprofloxacin and epidermolysis bullosa? It was somewhere around 18, correct? So when we say 18, that is well more than 2, correct? So it is a possible signal. Chi-square I can calculate there. And it is equal to or more than 3 cases. Yes, ciprofloxacin, we got 3 cases. So we can consider it as a possible signal. So why I am emphasizing this word possible signal? To say a possible signal, if you remember the definition of a signal, it should be a causal relationship. And we are not calculating any causality here. So because of that reason, disproportionality analysis is not a causal association. It is a hypothesis. The hypothesis is supposed to be tested with medical background or with any of the other means to confirm the signal. Okay, so please understand whatever research result you are getting out of a disproportionality analysis required to be re-evaluated with using a superior study design. So when I say superior, what I meant is say a case control or let's say a cohort or RCT, any of those things. Or maybe you can try for any genetic association or any of the in vivo or in vitro technique. Okay, so try to remember that. Yeah, so this is the two main common disproportionality analysis, correct? ROR and PRR. So this is the always confusing thing which to be used. So my personal suggestion, use both. So we should understand how they are different. When we say PRR, that is proportional reporting ratio, they will compare the drug of interest of the reporting rate of a drug in the class to the other class. But when we calculate ROR, what we will do? The event will be compared, the event of interest, with what is the frequency of the event of interest, and how it will be compared with the incident rate of the other event happening in the database. So when we compare PRR and ROR, the chances of false positive will be comparatively less. So my personal suggestion, or out of my experience, what I am suggesting is use both. If possible, use IC also. That is very good. But if it is not possible, at least use PRR and ROR. Okay, so there are few biases which can happen. Let's say, take an example. UITs related to rifabutin. Almost 75% of the report is because of UITs with rifabutin. So what will happen is when you calculate the signal strength of rifabutin, usually you won't get. The reason is 75% of the rifabutin is this particular event. So what will happen? The other result will be the other signal associated with the rifabutin will be reduced. Okay, so this is another error which usually happen. And in case of ROR, if either of B or C is 0, what will happen? You cannot calculate this particular signal. Okay, so this is a problem. That's the reason why I'm telling you, please take both ROR and PRR. Yeah. Moving ahead, which database can be used? See, you must have heard many databases in yesterday's classes. But when I was starting my research, maybe somewhere in 2014, we don't have access to any of the VGFlow or any of these databases, PvPI, any of these. So I was looking for a publicly available database which anyone can use. My search ended in FACE database. So you should understand one thing about FACE database. This is reported by medical professionals and the consumers. That means patients also will report. So there is a chances that there will be some spurious reports. So you should be skillful enough to identify which are the spurious reports and which are the actual good report. Yeah. So what is the advantage of FACE? It has more than 100 million report. Yes, you heard it correct. It's more than 100 million report. So it's a huge collection of data. So what is the advantage if the number of reports increases? Usually when the number of reports increases, the chances of getting false signal also will be reduced. Yeah, so this is the basic anatomy of FACE database. We have a drug file where you have all the reports of all the information about the drug. We have a demographic file where you have case number, age, patients, details, outcome, like what is the outcome of the particular event, therapy, which type of drug they used, indication on what basis, for what indication they have used, what reaction we are using, the patient experience, reporter source code. So all this information is available in the database, and everything is connected with the ISR number. So these all are different files, and everything is connected with the ISR. So previously, I started using Excel to connect all these things. It is quite time-taking, time-consuming thing. Later, there are many online portal available to doing this data mining. Later, I moved to start using R software for data mining, but I find it very easy with one online platform. So you don't have to know any of the coding skill. You put the drug name, put the event name, you will get the signal. I hope most of you are looking for such kind of things, isn't it? I'll introduce you after this particular theory session. Yeah. So what are the other publicly available database? Definitely, I'll ask you or urge you to go ahead with the VAERS, that is Vaccine Adverse Event Reporting System, Vigilance and Canada Vigilance Adverse Drug Reaction Online Database. I have personal experience with these two. I have published articles also on FAIRS and VAERS. Vigilance and Canada, I didn't go because I felt these two can serve my purpose. So if you are someone who is looking for a primary research, please go ahead and explore more about FAIRS. There are nearly 3,000 articles associated with FAIRS and nearly 1,000 articles associated with the VAERS. Please read few of them, okay, to get a better idea. So next question: do we need a what do you call a separate axis for using FAIRS database? Answer is no. The same question were asked, and I have sent a mail to FAIRS, and they replied that you do not need to require any permission. You can straight away go ahead and use it. Okay, so you are free to use FAIRS database. Okay, the ultimate question: how to frame a research question? So which drug event combination to be selected, or where we'll get the information? How much time it will take? Correct. I hope during the course of this presentation you will get answer for all these things. So see, the time and type of question is always depend on the study objective. The basic thing, or you can start with, is a novel adverse event of a drug. You can take up any of the drug, any drug. I'm saying any drug. Preferably, it has got it approval at least by 2019. It's not because of COVID. I'm saying 2019 because make sure that since it is available in the online platform, I want to tell you that whenever you are seeing this video, whichever year it is, take at least 2 years before that. So why 2 years? Because you will have 8 quarters of reports. That will be sufficient enough to generate the data. If you're taking a drug which is approved yesterday, the reports of that particular drug will be maybe 0 or very less in the database. So take a little older drug, like 2 years or 3 years before, or maybe you can take the drug which is approved in 1950 also. Okay, yeah. So what do you mean by novel adverse events? So as you know, the drug profile, when any drug, for that matter, you take any drug, and type prescribing information, you will get a particular PDF, maybe usually from the FDA website or from their the sponsors or the client, sorry, the sponsors website. Yeah, so you go ahead and read about the adverse events and clinical trial data. So they must have listed a few of the adverse events. Any adverse event you are observing beyond that, that means any new information you are getting, that is a novel adverse event. Another method is, I hope most of you will have micrometrics access. If you have micrometrics access, what you can do is go ahead and see micrometrics. Any report or any adverse event which is not mentioned in the micrometrics, that also can be used as considered as a novel adverse event. Okay. Or another method is a specific event by different drug, like suicide ideation. The drug which can cause suicide ideation, or outcome based, like the drug death associated with a drug or death associated with a vaccine, or a population specific adverse event, like you want to know human papillomavirus HPV vaccine infection among HPV vaccinated population. That also possible with the VAERS. Sometimes multi-stage studies also possible, like ischemic colitis in rheumatoid arthritis patient receiving TNF alpha inhibitor. Correct? So 3 stages. Medical devices also possible, like intrauterine device displacement with respect to LNG, that is levonorgestrel IUD. Then medication error or dosing error. So all these things are possible. So the first thing is you should understand where you are looking into and what you're looking into. If you have a clear picture of that, from there you can use any of the databases. What is the resource? Where you will get this information? So usually, from the hospital you will get this clinical the research question, or with a keen observation. Articles, if you keep reading articles, that is the next source of information, case reports or textbook. Yes, so enough of all this theory, let's do it. Before that, if you have any queries or any doubts with respect to the presentation, I will take up the question now, then we'll go ahead with the explaining the online portal. Krishna, any of the questions?
Bish, I think there are a couple of questions we can address before going to the live session. Yeah. The first one is what kind of research we can do by using this data?
Yeah, so I hope that the question must have come before the last slide. You can see this is mainly a hypothesis. We are trying to identify hypothesis. We are saying that I'll give you a few of example of which of my publication. Like I have conducted a study to identify the novel adverse events of vortioxetine, which is an antidepressant. Then brexpiprazole, another atypical antipsychotic, and their novel adverse events. So when I say novel adverse event, the adverse event which I identified is not there in the drug monograph. So that is one way of doing. Then few of my students, I cannot give you the research question because that is yet to be get published. They identified, they selected, let's take this example, suicidal ideation with respect to Montelukast. That is one article which we presented in ISPI. So that is another way of doing. You can identify an event and which all drug can cause that event. That is one way of doing. Other is you take a drug and identify the complete safety profile of the drug. That is another way of doing. Or if you want to be more specific, like a specific population, like say rotavirus, that is the one which I stopped in between. Rotavirus vaccine you give, but there are many cases rotavirus vaccination, cases which is being reported. So if you want to know how many rotavirus infection happened for a rotavirus vaccinated population, that also possible to do. Yeah, Krishna, that's all.
Yes. I think this is a question from Sweta Kumari. So I hope you got some understanding by this time. So we can use this data particularly to see any association between a drug and the reaction, which was not identified earlier.
Yeah.
So I think basically you can understand what are the reactions that are already identified for any medication. Maybe you can get from various sources. What are the aids already identified?
Yeah. And the sources can be like micrometrics will be the very easy source for all the students, or you can use drug monograph or prescribing information or FDA website, any of these.
Exactly. So wherever you find any reactions which were not identified earlier, you can take up that reaction and you can go for data analysis. Then you can prove yourself that is there any signal identified between the drug and reaction. So that's where we need to start the research. Yes. And one clarification from my side, don't confuse between yesterday's values, what I shown, and what's shown by Dr. Subis today. especially for ROR and IC values. The standard values, what I mentioned earlier, I mentioned we are going to be considered the lower limit of this ROR and lower limit of IC value. And even Subish mentioned same, but it is in a different format. Yeah, so, Subish mentioned ROR with the standard error and IC with
1.96 SC is standard error. That is nothing but lower bound value.
Exactly. So don't confuse. It is just a lower bound of the ROR and lower bound of the IC value. That's it, I think, Subish. Maybe you can go with the demonstration session.
Yeah, sure. So, in yesterday's session also, I have keep hearing one word, false signal. So, that is something which I'm going to cover now. Yeah. Okay, before that, I'll show you how to go ahead here. You can use this particular keyword. You will get here, go here. Yeah, I hope you will be reaching this particular window. Krishna, can you help me here? Is it visible?
No, Subish. We can still see your presentation only. I think you need to reshare once again.
Yeah. I'm so sorry. I'm sharing my screen. So
entire screen while sharing. If you share,
I'm sharing my full screen. This is my personal laptop, so apologize.
Yeah. Because that's the reason there'll be some problem.
Yeah.
It is visible now. Open FDA.
Yeah. So you can use this particular keyword, OpenFDA Shiny App Drug. So the second one you can take, you will reach here. Okay. So here what we are trying to explain is I'll start with one of my personal experience or one of my research which last year I published. The example what I'm using here is letrozole and osteonecrosis of Joe. Why I take up this particular example, because this is the query which I keep getting through mail of most of researchers working on this area. So I thought I will just make it clear, because it will cover both aspects like disproportionality analysis and false signal. Okay. So what happened is it was during my our usual ward rounds, and we have come across a case, a breast cancer case where the patient being diagnosed with osteonecrosis of jaw. Okay. So yeah. So we checked the monograph of letrozole, and we identified that as if now there is no report of letrozole associated with the osteonecrosis of jaw. So what we'll do, we will go ahead here. Yeah. See, this is the portal where you can click here and select the drug. Be very careful when you are entering the drug name. Okay. Yeah. So letrozole, depending upon the number of event or frequency you want, you can increase the number. For a time being, I'm not keeping very high number. For the ease of analysis, I'm keeping only 50. So what will happen is top 50 result will come. You can keep 500, but only problem is it will take some more time. So you can see that they are fetching the data of top 50 report. Yeah. So letrozole, the highest is for breast cancer metastasis to bone, metastasis to liver, neoplasm. Yeah, here we go. So there are 575 reports, where letrozole associated osteonecrosis of jaw in phase database. But when you see letrozole monograph, okay, I'll just show you the monograph also. Yeah, so here you can see that there is nothing like osteonecrosis of jaw is mentioned here. Correct. Even the rare also, it's not there. Okay, so you can just go through it. I'm not going very detail about it, because since it is very easy to see it. Yeah. So what is our observation? Where is it? Yeah. So we understood that there is nearly 575 cases of osteonecrosis of jaw, and letrozole is not known to cause. I'm giving it more emphasis. Not known to cause osteonecrosis of jaw. So as a researcher, I'm very happy that I got something very novel, and I start searching more about it. Okay, so if I go inside and see, if you click on the number, you will be getting the individual cases where you can see each and every case. You see here, you can see each and every case. What you can do is go to the next report. You will get the next report. They are giving letrozole, tamoxifen, bisphosphonate, denosumab. Correct. Go to the next case here. You can see that what are the drugs they have taken. You got me. So you can go ahead and see all these cases. But this is not possible if your number of cases is like some 10,000 or more than that. Practically it is difficult. So that is somewhere which I got stuck. Then I was keep searching and got an idea about Open-Vigil. So this is another online portal which I want you to have a look. Okay, so Open-Vigil, it is developed by Mr. Bo, and the access to it, you can send a mail to him, or maybe you can drop a mail to me. I can share you the user ID password. Since it is an online platform, I cannot share it. You can contact him, and please make sure that you give enough credit to him, okay, when you use this particular portal. Yeah. So you will have all the information here, how to use Open-Vigil. You can go ahead and see here. But now what I'm trying to do is, yeah. So what we are trying to do, we have a research question. What is our research question? Letrozole and osteonecrosis of jaw. So here disproportionality analysis, significant association between condition 1 and condition 2. Okay, so this is what I'm clicking. Go down, you can type here letrozole, osteonecrosis of jaw. Yeah, you can download it as a CSV file, or you can download it, you can view it also. I'm giving the view option. And this particular, see, I'm using 100 Mbps net, so that's the reason I'm getting fast. So if you don't have a very fast internet, it will take time. Okay, so make sure that you have a good internet source for doing this study. Yeah, so what we got here, we have relative reporting ratio 11, PRR 11, ROR 11, correct, everything. IC is 3, very promising result, correct. But one more thing I want you to remember is, you should always look the co-administered drug. See, so what are the co-administered drug? You can see letrozole, denosumab. Out of 500, around 675 cases, you have denosumab in the report. Correct. Then exemestane. You can see all the zoledronic acid and bisphosphonate, etc. So why I'm emphasizing this? Because when you do a study, after you got a signal in Shiny app, please come here and search for the co-administered drug. Then please copy all these drugs and keep it in Excel sheet. And what you should do, go ahead and see whether any of these drugs can cause an adverse event. You understood? I'll repeat. We have a hypothesis, or we are trying to identify whether letrozole can cause osteonecrosis of jaw. But there is a possibility that some drug which is given along with letrozole can also cause. Correct. So we call something like innocent bystander. So to eliminate that option, or to eliminate the concomitant use of other drug causing the signal, what we will do? We will try to unmask the signal. So how to do that? Identify all the co-administered drug, and what next? you see, you make a list out of these drugs, how many of them are known to cause osteonecrosis of jaw. Okay, so let's say you got two drugs. Yeah. So this is open FDA 2.1. So you got two drugs. Say letrozole. Okay, before that, I'll just run this query. Yeah. So you know that denosumab. This can cause osteonecrosis of jaw. Okay, so what we should do? We should eliminate all the reports of denosumab from the reports of letrozole. Okay, so let's say you have 100 reports of letrozole. In that 100 reports, 50 reports, denosumab is co-administered with letrozole. So there is a possibility that in that 50, the report is because of denosumab. So what we can do? We will remove that 50. We will take only the remaining 50 where letrozole is administered alone. I mean, it's not with denosumab. Okay, and there is one more drug which can cause, that is zolentronic acid or allantronic. Allantronic or any of the bisphosphonate. Okay, I'm not putting the complete list. What you should do is you keep your drug of interest here, and which all report you want to remove, that you keep as and not. Got me? Yeah. And here you can see it will be usually in LTT, that is lower level term, that you please change to PT. That you please change to PT and type your event of interest. Osteonecrosis of jaw. Okay, yeah. So you have subgroup analysis here, which I will be showing later. So what is the result you are expecting here? See, after doing this study, after removing the denosumab and alendronate report, we are expecting a positive signal. Okay, let's see what will be the result. Okay, let it run. Then another thing what I want to tell you is subgroup analysis. So this word also you must have heard yesterday. Let's say, see, I know that letrozole can cause osteonecrosis of jaw. I want to know which gender it is more susceptible. So what I'll do, I will keep male here, and oh, I made a mistake here. I think I only filtered for male. Okay. I think one is sufficient. Okay, that let it run. Yeah. So you want to do a subgroup analysis. So I want to know letrozole and osteonecrosis, but who it is more susceptible. So what you can do? You keep your drug of interest here. You keep your event of interest here. Then what next? Go here and put male. Okay, and put the search bar. Let me close all other window, otherwise I will get confused. Okay. Then what next? You want to see for female. Search. Yeah. Then what else you want to know? You want to know whether it can cause any with respect to age, it has any impact. You want to know who having more risk, whether adult or post-menopause. Okay. Sorry, yeah, pre-menopause and post-menopause. So who have more risk? So we gave here search, come back, 46 to search. Yeah, I think few of our result already came. So you remember we gave letrozole, we removed denosumab, we removed alendronic acid. What happened to the signal strength? So signal strength before it was 11, it dropped to 6. You understood? So if you keep all the bisphosphonate, the signal result will be 0.1. Okay, what is happening here is, you should understand, this is very important. For what we use letrozole? Letrozole is used for the treatment of breast cancer. And when you say breast cancer, that always metastasis to bone. Correct. And in bone metastasis, we give denosumab or bisphosphonate. Correct. So it is very common to cause osteonecrosis of jaw when you use denosumab or alendronate. Got me? So this actually, here letrozole is an innocent. It is not causing any osteonecrosis of jaw, but the drug which is co-administered with the letrozole is causing all this issue. Got me? So don't ever go with open FDA report alone. You should do a subgroup analysis. You should analyze the co-administered drug. Maybe you can put a regression line and check it, but I cannot show it here because of the time constraint. You can do a little bit of coding for that. Then another method is removing all the reports of co-administered drug. Same thing what I did here. Okay, yeah. Then letrozole for male, yeah. So we have 3 reports of male. Yes, breast cancer can cause for male also, right? It can be male also. So male, the report is, the PRR value is 6, and for female, the PRR value is 8. So we can say that female having more risk of osteonecrosis of jaw. Then we compared age group, correct? So this is, oh wow, you see this result. When we compare the age group of 19 to 45, their signal strength is 47. Where when we compare the signal strength of post-menopause, see their signal strength is 5. So what is the inference we are getting from here? We can say that during the reproductive stage, the chance of osteonecrosis is high associated with letrozole. Correct. So like that, you can do many things. See, this is an example where I kept age. So instead of that, you want to see the outcome. So you want to see how many of them got congenital anomaly. That also possible. Or you want to see, how many of them were COVID-19. That is a recent trend, right? You can check that. Or you want to see which country, you want to compare US versus UK or India versus UK or Europe. Europe is not possible. Any of the country you can compare. Okay, so that is another subgroup analysis what you can perform. Yeah, one more thing I want to tell you is you can download this particular thing in CSV file, Excel CSV or HTML file also, where you can download it and do for further analysis. Okay, yeah. So I think that's all I can do here. If you have any question, I can further extend the talk. Krishna, can you help me here?
Yes.
If anyone have any doubt, I can explain it further.
I think very good information we have given already for them. Mm-hmm. And in another 5 minutes, maybe we can discuss a few things. So it is going to be very clear for the audience.
Yeah.
And
see, what I'm personally suggesting is you please go ahead and explore this OpenVigil. It is very simple, user friendly, and Mr. Bo is very helpful person. Only thing you have to show little patience and make sure that you give enough credit to him when you publish. And if you have any doubt, you can always contact me, or in fact Krishna also working in this particular field.
Yes, Subhesh. So for the benefit of the participants who are here, maybe we can do a few things so that it is going to be more clear for them. Sure. I think one thing not clear here is from where the data is coming to OpenVigil.
See, this is actually running, on Python platform where, the data is coming from FAIRS only, FDA database only. So you remember I shown you this. This is OpenFDA where the data is coming from FAIRS database. So this is one portal. The same thing, they're doing little advanced analysis using OpenVigil. So advantage of OpenVigil is there are a lot of duplicate report. I'll tell you an example what I mean by duplicate report. See, as a clinical pharmacist, you must be reporting it, correct? And as a mandatory reporting, the sponsor is supposed to report it. And let's say there is a pharmacology, MD pharmacology doctor who is also reporting this. And let's say the nurse is also reporting this. So from the same patient, maybe 4 or 5 reports will be going to the database. Correct. So there will be duplicate report for the same thing. So that can be avoided in OpenVigil. OpenVigil is a cleared cleared database of affairs. And see, as we know, we need age, gender, indication, drug, duration is mandatory field for report. So that will be make sure that all those reports alone will be given in the OpenVigil. The remaining reports will be removed. So when you search OpenVigil and OpenFDA, you can always see that OpenFDA will have more number of reports and OpenVigil have less number of reports. The reason is OpenVigil will remove all the duplicate.
Yes, yeah. I think for the participants now it is very clear that OpenVigil is just a platform. It is not having any areas. It is just taking all this information from FDA database, that is FDA AERS. And by using this platform, we can do various kinds of analysis very easily. And Subish, you have shown two things. One is OpenFDA, another one is OpenVigil, right?
Yeah.
So these two are two different things, or they are both related?
It's like Amazon and Flipkart. The purpose is same, shopping. The background database is same, that is FDA. Only thing, we have two type of analysis. Both are doing the same analysis only, but only difference is that is the crude database what they are using for OpenFDA. They won't remove any of the duplicate, any of those things will not be removed. So on a primary analysis, you can use OpenFDA when you have a confirmed signal or you answered your signal or your research question. Then you come back to OpenVigil and do the advanced analysis here. The subgroup analysis, removing the concomitant drug, all those things you can do here. See, what I shown is only 10% of OpenVigil. You can extend this class for maybe one day also, because it is very vast. It is very difficult to concise into one class.
Yes. And Subish, I think it will be clear if you can explain more on this particular aspect.
Yes.
Nowadays, actually earlier, whatever the data you want to have from FAERS, we need to download the Excel sheets and to go through. I think downloading itself is going to take 1 or 2 days time, right?
Yes.
They are very big files.
Yes.
And now FDA itself is come out with the open platform where you can go and search for the information. I think it's an open FAERS we are having.
Yeah.
So there you can go and search for the medication and the reaction. We can get all the details, right? So compared to the original FDA open database, what are the advantages we are having in this OpenVigil? I think you already explained, but maybe if you
Yes, I got your question, Krishna. So, older days for getting this result, it will take at least 1 month. That much of hard work we have to put. Now, within, I think, half an hour, we at least explore some 3-4 signals. That is the advantage, main advantage. Time can be, with one click. So basically what they'll do is, I can show you the query. See, this is sending a JSON query to the FDA. See here, this is the query what they're putting. Actually, you're supposed to type it, but because of the advancement in the technology, you don't have to type any of these things. You just type your drug of interest and event of interest. They will do the coding part and they will send you and fetch you the result and give it to you. So that is the main advantage here. Then second thing is, sometimes towards that you will understand that you are following a wrong research question. So all your hard work will go in waste. So that also won't happen here. You will get to know well in advance. So my main thing is the error will be comparatively less. You will get a more refined data, more reliable calculation. Since it is completely done by the computer, you can more trust those calculations rather than human calculation. So that's what I think, Krishna. It is
exactly
it's quite easy. Anyone can do it now.
Yeah. OpenVigil gives, I think, all the types of the advantages for us. because we can do
ROR, PRR, everything will be given, and subgroup analysis also will be given.
Exactly. And
one
more
thing which
important,
I'm missing in OpenVigil is time to onset, like how much time it took for the event to happen. Those information is not possible to do with OpenVigil. That you have to use either R or Python or any of those softwares. Softwares
yes,
means data mining method.
the very important aspect here it is what you mentioned, the signal refinement. Of, eliminate all these confounding factors and other covariates which can also cause a reaction.
Yes.
Doing this signal refinement analysis so that we can have very strong signals which are not false positive. So I think that's maybe the biggest advantage.
The reason why I'm emphasizing this, because see, out of interest, the students or whoever, the researcher, they will go ahead and do this research and they will send to some journal and they will get rejected. They will ask you for all these questions and you will feel bad that you put all your hard work and get rejected. Rejection is not something which we want, right? So you please make sure that whatever signal you are getting, it has some value or it makes sense. You ask yourself, what is the possibility that this particular event is happening? You should identify what is the mechanism by which this is happening. So all these things are very important. Maybe you can identify a genetic factor associated with this, or you can do a docking study to identify the association between the receptor and the event. All these aspects also can be explored.
Yes. Thank you very much. Subhish, one last thing. We have one speaker here, very senior person from industry in Pharmacovigilance, Dr. Mohinton. He raised his hand. I think so. He's having some question.
Sure.
Yes, thank you so very much. In fact, I was not aware of the program that you are going to speak on Signal. Otherwise, I would have joined much earlier. I just heard the late part of your OpenVigil. I was very curious whether this OpenVigil is available to marketing authorization holder companies, or this is a private tool which people like WHO and does UMC and PvPI use this tool, or they don't do it?
No. This is actually developed by a researcher called Baum.
Okay.
Which is published in British Journal of Clinical Pharmacology in 2021. So this is more of a research tool rather than a tool which can be used for a sponsor or any of the organization.
So if I want to use this, how can I make use of this tool?
Sir, it is freely available.
It is freely available.
Yes, 100% free.
That's great.
Yeah.
I mean, the type of I just saw the way you were searching. Pictures are fantastic, and within 5 or 10 minutes time, if you can get the PRR value and all. So it made great interest, and I think thank you so much for this, and I would definitely go and use this tool myself. Thank you so much.
Please, thank you.
It is just like a PubMed, sir, where we can go and search, keep away and or not, so that we are going to get all the information. I think it is based on the interest the researcher developed, so that many people started using it.
Mr. Bo, Dr. Bro, because of him, we could able to do it.
Thank you so much. Thank you so much.
Thank you, sir. Thank you for your
for wonderful explanation. I'm sure that this is going to be the reference video, especially if someone want to see this OpenVigil in the future. Thank you very much.
Yeah, I shared my contact details. If anyone have any doubt, you can put a mail to me. Avoid calling.
Yeah.
I'll be answering in the text. Anytime you can contact me, I'll be happy to help you.
Sure.
I will be the first person to write to you.
Please, sir, please. Okay. It's a privilege.
Sure.
At this time, they understood that you are an open person for OpenVigil. So,
yes.
Thank you.
Yeah, that's great. Okay. Thank you so much.
Thank you, sir.
Thank you, Subhish. Thank you for joining with us.
Thank you, Krishna.
So hope the audience enjoyed the session, the first session of the day. And with this, we will go to the next session. We have a very experienced speaker with us in terms of pharmacovigilance. We have here Dr. Mohinton, who is the CEO and founder of PVCon Pharmacovigilance, Auditing and Consulting Services, Mumbai. And you see the SARS experience, I think it is not, the time is not sufficient to read everything. That much credentials he's having in terms of pharmacovigilance. He's having global experience in handling drug safety surveillance activities in both pre-marketing and post-marketing arena, which includes all the activities in terms of pharmacovigilance like protocol assessment, starting from safety point of view, preparing medical as well as safety monitoring plans, safety monitoring and documenting, including EDCs, assessment and regulatory reporting, including CSR and line listing. And also he is in conducting various post-marketing surveillance studies by using registries and handling ICSRs with a complete processing, assessment, quality, medical and regulatory check, reporting generation, as well as submission to the marketing authorization holders. And handling and maintenance of databases, for example, PV software such as ARIS Global, electronic submissions, and PSERS, signal generations, as well as risk management. Sari is having vast experience in the area of pharmacovigilance, and I can say that Sari is the Moin Dawn is a dawn for pharmacovigilance. That's a very simple sentence to explain everything. So thank you very much, sir, for joining with us to explain few concepts on pharmacovigilance to the audience. Now the session is yours.
Can you share my slide deck? I'm having some problem here so that you can navigate through.
I can do that, sir.
Yeah. Thank you. So good morning, fellow victims of COVID-19. I think we all of us are affected directly or indirectly by COVID-19, and you would agree with me. If this situation was not there, probably we would have been meeting in person, face to face, and I would have been showing you this presentation to all of you. So I feel sorry for myself that I cannot really come in contact with you face to face, because I always prefer that mode of teaching. First of all, I must apologize to you, because when I saw this today's presentation by Dr. Sadbir, I think he has taken a topic which goes much higher in the PV chain of events. You know, we start with ADR reporting, and finally we end up with signal detection, and then based on the signal we actually take a decision what needs to be done with a confirmed signal. So my presentation today would be like a nursery rhyme to a postgraduate student. So I'm so sorry. Because I did not have any idea of other presentation what are going to be made during this pharmacovigilance week. So if you find it very naive, or if you find it very preliminary, my apologies for that, for having misunderstood. And I really do not know how many of you are actually involved in day-to-day pharmacovigilance activities, how many of you are undergraduates, how many of you are postgraduate. But what I will do while discussing the slide deck, what I have in front of you, I will definitely try to get in more insight from my 40 years of PV experience in the industry, and also as a consultant. So I have been working now as a PV consultant for last decade, 10 years, and have a lot of big companies as my clients. So I'm fully aware of the various facets of pharmacovigilance starting from A to Z, and that's why I have put the title of the presentation as PV industry practice, part of the national pharmacovigilance. So as you know, when you are doing a pharmacy, B Pharm, or you are doing M Pharm, there are a lot of questions which come into your mind, what you are going to do next. I mean, we are talking about the career pathways. Okay, so whether, I mean, there are various pathways. Just go to this thing, next tab, and you will have. So you can do, graduates can do post-graduation, then they can do doctorate, then they can take up a pharmacy, or they can have their own pharmacy. They can take up a research job, like we have a lot of research jobs going on in drug safety. You can join industry, or you can join academics like what Dr. Krishna is there. Or you could be your own entrepreneur like I am. There could be a lot of other people who are doing their own business, which is associated with pharmacovigilance. So pharmacovigilance, sky is the limit. And I must say that today, pharmacovigilance is a hot potato. It is in demand. And as I will take you forward, that how much, I mean, I will try to give you some figures which will really help you understand the magnitude of demand of pharmacovigilance specialists and experts, especially in India. My lessons learned from this pandemic, I must share with you that we have come across healthcare professionals who were good Samaritans and who went out of their way to help people who were suffering during this pandemic, first wave, second wave, and God forbid we are expecting a third wave. That's what people guess. But at the same time, I have come across, I have been working at the ground level, and I have come across a lot of healthcare professionals who were behaving like bloodthirsty vultures, and they really, really exploited the situation to the hilt. And you know that how drugs were sold in black market, how oxygen was sold in black market, and hospitals charged them fees which was not possible for a poor man to afford. So my first, you can call it sermon, you can call advice from a senior professional like me to all my budding professionals, that it's good to be a good professional. It is extremely good to be an excellent professional. But most importantly, all of us should be good human beings. If our professionalism is not helping the humanity, then this professionalism is of no use. And at least we should not use our professionalism to exploit people in the name of healthcare. So when you think of industry, now I'm not taking the academics or I'm not taking to the research, because basically my domain has been industry. So if you talk of industry, these are the only few things which I have presented you. You can be part of the production units where formulations are being made, and you have a lot of opportunities in Chandigarh and Baddi and areas around it. Then you can be a part of the analytical team. You can be part of sales and marketing team. This is very essential for you people to introspect yourself, the type of personality you have. Everybody cannot can become a sales and marketing person. So if you are an introvert, if you are hesitant to talk to people or engage people, if you do not have the type of command over spoken English, then sales and marketing is definitely not your cup of tea. So you can think of production, you can think of analytical, where you are supposed to be working on your own. The newer horizons which are coming up now for pharmacy graduates and postgraduates is regulatory affairs and patents, then service industry like CROs like BPOs and KPOs like Cognizant and Tata Consultancy and Covance. And of course, you can be also part of the formulation development. So you have to choose one of these things if you want to come to industry. I have not taken medical writing into this. That automatically becomes part of the service industry, because most of the people are outsourcing pharmacovigilance services along with medical writing to big KPOs like TCS and Cognizant. Next slide, please. So, let me take advantage of the current situation, which is COVID-19 vaccine. Why there are so many questions and uncertainties about COVID-19 vaccine, which just came after the first wave and people started developing. So there are questions and question mark against the efficacy, because we have been getting a lot of reports that having taken 2 shots of vaccine, people still suffered. from COVID-19, and safety is, again, there are many questions against safety, because as I'm sure as a student of pharmacy, you must have known that whenever we used to talk of a new investigational drug, we used to talk that it takes something like 10 to 12 years to come up with a new investigational drug getting approved. So you must be wondering what happened to COVID-19 vaccines, which were developed and which were thrown into the market within 2 years' time. And as you know, that a lot of regulatory agencies have approved COVID-19 vaccine, even some vaccine where the phase 3 trials were not completed, and on humanitarian ground and compassionate ground, they have given the approval so that a larger threat is avoided. And as far as safety is concerned, just let's go to the next slide, probably where you will be able to understand where I'm coming from. If you see the clinical investigation, whether it is phase 1 or whether it is phase 2 or whether phase 3, all these three phases, you will see I have written there the first administration in the men, which is phase 1. So naturally, we are talking about tolerability of this new molecule in human being. Then in phase 2, again, we have safety as the primary endpoint, and in phase 3, we have efficacy, we have indication, and also short-term safety. So when we say of short-term safety in clinical trial in phase 3, there are some inherent limitation of clinical trial as such. So even if a company has gone through the entire phase 1, phase 2, phase 3 trials, and they have their dossiers ready and they have their efficacy data and safety data, still this safety data is considered as a provisional safety data for this particular drug. I don't want to be repetitive. I'm sure you must be knowing the shortcomings of clinical trials, that during clinical trials, the number of people who are exposed are very, very less number of people, maybe 5,000 or so, or the window period for which people have, or the patients have been tried, it is a very short window period where they are exposed to the new drug. Then by inclusion and exclusion criteria, there are a lot of human species who are not really exposed to this drug. So we don't include pregnant women or elderly patients or kids in this. So the clinical trial data, and especially the safety data, which comes out of part clinical phase 3, has inherent problems. And that's why after having a new drug approval, where you have already proved that the benefit of this particular molecule is overwhelmingly more than the risk provided by this particular drug, all the regulators give you a go ahead, and then you bring out this particular molecule in the marketing or into the public, where people who were not exposed to this particular drug are exposed. And that's why the phase 4 or the post-marketing surveillance becomes very, very important arena, where the risk profile of the new drug is further characterized, further it gets illustrated, and we have to collect data through various post-marketing surveillance methodology. The best one being used is your voluntary reporting, which is known as, everybody knows that 90% of the cases are coming to marketing authorization holder through spontaneous reporting. And that forms the crux, that forms the backbone of the safety analysis of all the molecules which are there in the market. And this phase. As you would understand, I don't know whether during your learning phase, during these 2 days or prior to this, I'm not sure whether you have ever heard the rule of three. So the rule of three says that if a adverse drug reaction, the rate of occurrence is 1 in 10,000, then to find out or detect or identify such adverse drug reaction, you need to have 30,000 patients enrolled or subjects enrolled. So you can imagine if a ADR, which is very, very rare, which is 1 in 1 lakh, you will have to enroll 3 lakh patients or subjects during this clinical trial phase, which is not possible. Even, 1 lakh, or if you ask me, so what I'm trying to say that if you want to find out or identify a rare type or a very rare type of adverse drug reaction, which will be caused by this new molecule, you really need to have a huge pool of data from where rare or very rare adverse drug events can be identified, highlighted, and they can be included in your PI, it could be included in your CCDS after having confirmed signal. So how do you get it? If you don't get it from clinical trial, naturally, you have to have a robust pharmacovigilance system within your company, and through which you will be collecting ADRs like what PvPI is doing in India, or WHO or UMC is doing worldwide. Similarly, every pharma company or marketing authorization holder, it's their obligation to have a very, very robust pharmacovigilance program. So when we talk of pharmacovigilance program, it does not include only the clinical trial phase or the drug development phase. It goes beyond that. Means after getting the marketing authorization, the company is supposed to continue to do the surveillance, the safety surveillance, till such time that the drug is there in the market. So this is your obligation as a MAH. This is my obligation as a pharmacovigilance professional working in a pharma company. And that's why pharma companies have very, very robust pharmacovigilance department and function, which I'm going to share with you. So, many people think that if a drug has become a mature drug and the drug has been existing in market, then it is presumed to be safe. Let me give you an example of paracetamol. Paracetamol, every day in and out, all of us have been taking paracetamol, thinking that paracetamol is very, very safe drug. But recently, FDA came out with confirmed signal paracetamol causing SJS. So we had never heard of paracetamol causing SJS, but now Stevens-Johnson syndrome has been a confirmed signal by US FDA. And US FDA has sent a communication to all paracetamol users, all physicians, that they have now confirmed signal that paracetamol can cause SJS. So what I'm trying to explain to you guys, that no drug is safe, first thing. Secondly, as long as you have your drug in the market, it is your ethical and it is your regulatory mandate that you would continue to do safety surveillance of your drug as long as that drug is there in the market. Hope I have made my point clear. So let's go to the next slide. So I'm not going to talk to you about pharmacovigilance. Just go to the next slide and the next slide. Yes. This slide is important. I don't know why I want to explain this slide a little bit in more detail, that this is a simple definition which has been given by Uppsala Monitoring Center. But here the 1, 2, 3, 4 red lettered terms are very important. So when a company like Sanofi Aventis or Johnson and Johnson, where I was working, and in Johnson and Johnson, I was the regional director for Asia Pacific for pharmacovigilance quality assurance. We had a huge team of pharmacovigilance. Our team, I will come to that little later to explain you what is the importance of pharmacovigilance to the industry, okay, and to the humanity at large. So can you go back to the previous slide, please, Krishna? So you cannot, for detection, naturally, you cannot detect until and unless you have infrastructure. You train people, you have infrastructure for doctors to, report, find out from the patient and report this. So all healthcare professionals have been trained by most of the pharma company. They train their MRs and then sales representative, intern. They train their prescribers, that doctor, in case if you have any adverse drug reaction coming because of our molecules, please you report it to us. So that's how the detection takes place, and detection does not end at detection. After having detected, this needs to be communicated to the marketing authorization holder. So this happens either through apps, or it happens through portals, or in India, mostly it happens through sales representative and marketing RMP, this thing, sales marketing representative. So they normally carry a form which gets filled. It comes to the PV department. They do the assessment, and then they understand what the problem is. So you can imagine that just getting the report is not enough, like what you were seeing in the assessment open vigil. You really need, so you can imagine a huge big company like J&J or AstraZeneca or Pfizer's. They are getting thousands and thousands of ADR in a year. So what happens in case if they just get these ADRs and then they don't do any analysis on that? Naturally, they will not have, they will only have data, and they will have no information from the data. So to get information from the data, they need to assess that how many cases for this drug, how many cases for this drug, how many are serious, how many are not unserious. Then they'll go for causality assessment, whether there is a causal association with this drug, with this particular adverse drug reaction. So they understand what type of reactions or adverse drug reactions are being caused by our drug. And depending on this, then as you were previously told, that they have signal detection, and in case if they have any confirmed signal, then they need to take some regulatory actions, which I will come to my slides, forthcoming slides. But I just want to bring to your notice about drug-related problems. I don't know whether you heard about drug-related problems. This is an addition which has come after 2012, when European good pharmacovigilance practices came out with Module 6. And in Module 6, before this Module 6 coming up, we were just concentrating on pure ADRs. So, if a patient has taken a drug for a particular ailment or disease, and if it causes any abnormal, unfavorable sign, symptom, or disease, which was unexpected, then this is known as purely as a adverse drug event or adverse drug reaction if the causality is proven. But now they said, oh, okay, fine, this is fine. But we would also like to know what happens to the patient in case if your drug is given. So whether there are instances of drug misuse, drug, what happens, how your drug reacts during the pregnancy or breastfeeding or medication error. There are instances of LOE, that is lack of efficacy. So now the pharma company is tuned up, and they have refined their training, that besides capturing the pure, adverse drug reaction, they have to also capture what is known as instances or incident of special interest. So all these 11 circumstances or episodes starting from drug abuse to medical medication error to lack of efficacy, pregnancy, breastfeeding, and all these are to be also taken care of. So when even if they are not associated with an adverse drug event or reaction, for example, if a person has been given a wrong dose because of medication error by a healthcare like a nurse, so this would be reported. Doesn't matter even if the higher dose given to that particular patient did not have any adverse event, but this needs to be, because the regulator wants a pharma company to take cognizance of this, that wherever there are chances of medication error because of look-alike formulation or presentation or similar rhyming names. So the pharmacist dispenses something else which is similar looking name instead of the actual medication which was written by doctors, and you know how good handwriting doctors have. So, regulatory agencies would like that the pharma company should take cognizance and they should try to avoid all such circumstances where medication error is possible and which could be harmful to the patient. Next slide, please. So, you can go to the next slide, Krishna. Yeah. So I don't want to go into this, but this is just go to the next slide so that I can explain you much better what I'm trying to explain it to you. Yeah. So this is, in one shot is pharmacovigilance. So right from the phase one till you are 60 years plus in the market, this is your goal of pharmacovigilance. You have to continuously evaluate the data which is coming from various stakeholders, which is coming from clinical trial, which is coming from BAB studies, which is coming from pharmacists, which is coming from patients, consumers, their relatives, it is coming from health authorities, and whether it is coming from spontaneous, event which have been reported voluntarily by patients and patients' relatives or doctors. So you have to have a system in place, a very robust system in place, so that you continuously keep analyzing all this data which is coming to you, and you make from this data derive information and inferences, and through the inferences you come to some confirmed knowledge about the safety and the efficacy of your drug, and that's what you prove when you write a periodic safety update report. So you must have heard about PSUR, or now this is called as PEBR. So what do you do in a PSUR or in a PEBR? So in a PSUR, which is known as periodic safety update report, which has been now made and where benefit also has been added, every marketing authorization holder has to prove and submit this data to the regulatory authority of each and every country where their drug is registered. Remember, registered, even not even marketed. Even if it is registered, they have to submit at a predefined periodicity this particular data in a particular format where you describe and analyze all the information which you have for this particular molecule, maybe for 6 months, maybe for a year, maybe for 3 years, or when it goes further, then it could be a 5-year report in which you would try and convince the regulatory authority that, look, sir, the benefit offered by this particular molecule of our is still it outweighs the risk which is there associated with this particular molecule. So the benefit risk ratio for this molecule is still positive in favor of our molecule, and that's why we would like to continue to market this molecule in the market. And besides this, in case if you have some risk which is there on the left hand side, then the marketing authorization holder have been asked, or they out of their own prepare a risk management program. Okay, so the which is known as RMP. The risk management program is our plan is a very highly scientific and medical document through which you discuss and which you show your entire plan to the regulator that we have identified risk with this particular molecule, and these are the potential risks with this particular molecule, and these are the risks which are unknown about this, and what we are going to be doing to get this particular risk known, and how we are going to manage the already important risk for this particular molecule. So let me give you an example of thalidomide. When thalidomide was banned in 60s, now thalidomide has come back to the market for solid tumors. So why? Because the risk benefit of thalidomide now has totally changed. In 60s, the risk was that it was causing phocomelia, and the benefit was only that it was used as an anti-emetic drug. So the risk was much, much more than the benefit offered by thalidomide in 1960s. Now, in this century, our 20s, what we said that the thalidomide is being used as a life-saving drug for cancer patients for their solid tumor, and the risk of phocomelia has been totally taken care of by having a risk management plan whereby the thalidomide is being restricted, that it is being sold by or it is given administered by a onco physician, and this is not administered to women who are in their reproductive phase, so that the risk which was there for phocomelia is totally eliminated, and this becomes a life-saving drug. So its benefit really outweighs the risk now, and that's how companies and marketing authorization holder continue to do surveillance of benefit and risk and produce this document in so that they can have. Otherwise, if the regulatory authority is not really convinced with your documentation, the type of data what you have provided, if they still feel there is the risk and risk is not being taken care of, then they may cancel or revoke your marketing authorization, and it will be great harm and loss of monetary loss to the companies. That's why they had to be extremely careful while writing this particular document. And to write a document scientifically correct, you need to have a lot of data, and that's what you have. I'm going to show you in the next slide, please. So, we talk of PV, but PV is not a simple, a one, what should I say? Okay, it is not a simple terminology like this. I mean, when we talk of PV, there are many aspects of PV. And I'm not sure about the smaller Indian companies, but all big pharma companies, including Indian multinational companies, they have so many departments. So right from the highest level, they have a safety monitoring board, and they have a qualified person for pharmacovigilance. They are the highest authority which have been authorized by the company management board to take any action which is suitable for. So if they want to recall a drug, or if they want to make a change in the CCDS, that is the the core company data sheet, which is like the Bible, safety Bible for that particular molecule, or if they want to make a change in the PI or in the investigator's brochure, then this decision comes from the highest authority. So then they have a department which is known as end-to-end case management. So when we say, I'm sure that you must have heard about case management. So I really do not know how much you know about case management. It is the most labor-intense work in pharmacovigilance chain. Okay, and as you must be knowing, I'm just giving you an example. Probably this would be beyond your imagination that big companies like Roche and Johnson and Johnson, and I don't think there is any company, because according to a survey recently which was carried out by a very reputed company, that all over the world 70% of the PV work is being outsourced today by big pharma companies. And out of that, the case processing is the number one work which is being outsourced. And the most privileged countries who are doing this outsourced work is India and to some extent Philippines. So in India, as I will come to you, I will see in my some of the slides. Today, India is doing 80% of cases being processed worldwide. So 80% of ICSRs are being processed in India by Indian KPOs like TCS and Cognizant and Cyphomics, which has become now Covance and Accentures and so on and so forth. So I was just trying to give you an example, magnitude, that to what extent, how many people are being used. So if you take a big company like Roche or Johnson and Johnson and Pfizer, if you see their bench strength over there, that the number of people which are being used to do the case processing of only one pharma company, one big pharma company. A KPO uses something like 600 to 800 FTEs. That means there are 600 to 800 people working every day, round the clock, 365 days, only to do case processing of one company. Okay, so you can imagine that how many people are required, and this is the most employment generating segment in pharmacovigilance, where a pharmacist, where a medical graduate, where even a life science graduate is being taken as a safety associate, a safety scientist, who start at the very basic level of case processing. Then they go on to do the quality check of the cases which have been processed by data entry people. Then we require what is known as medical assessment, where doctors are required to do the causality assessment, the labeling assessment, the seriousness assessment, and to decide whether this case meets the criteria of submission to the regulatory authority, and then they go and submit this particular case. So this this is one particular division, a subdivision of pharmacovigilance juggernaut. Okay, then there is a very, very important, no, I have not, I don't know whether we will be able to complete. So let me just go through this, and then probably in case if we have little more time. So we have then literature search. So you know that there are a lot of literature available, medical literature, where doctors and scientists, they do a lot of studies, and then they don't report this to the pharmaceutical companies or even to regulators. They publish this as the papers because they would like to have this paper under their CV. So the regulatory authorities want or the MAH to do literature search once in a week to find out if there is any literature report which is associated with their molecule and which has a safety associated episode. So you have to also take care of that. Then I talked to you about aggregate reports, why these aggregate reports are important, because they give a worldwide safety review of the data which has been collected over a period of time which is predefined. So you have to have people who would be continuously working aggregate reports, because there will be some molecules where you have to give a 6-month report, there will be some molecules you will be giving yearly report, and so on. And there will be so many countries. Just you imagine that big companies like Johnson and Johnson and Sanofi Aventis, we had more than 100 affiliate countries. So it is the requirement of the headquarters of pharmacovigilance, where more than 300, 400 people are working to ensure that all aggregate reports are being sent to the regulatory authority of these 120 countries where this their products are being registered. And not only aggregate reports, wherever there is a qualification that a ICSR needs to be reported within 15 days when it is a serious adverse drug reaction, then, and which is according to now new EU regulations, irrespective of labeling assessment, this needs to be reported within 15 calendar days to all European states, member states. So it has to go to Vigilance. So the timelines are very strict, and then if you don't do this, naturally you come into the eyes of the regulatory authority. They can come for inspection, they can even revoke your license. So similarly, aggregate reports, if you are not submitting in time and if these reports are not really very technically correct, then you can have queries, and then your company can be in serious trouble. Then you have signal management. That's what in the previous episode we saw how signal management is done. So there is a big team who is doing signal management, and they are all doctors from various therapy areas because they understand their therapy area well. Then you have risk management plan. That's what I talked to you in the beginning, that you have a risk management plan, and according to the risk management, in case if there are changes to be made in the label, or in case if you have to issue dear healthcare professional letter, then this is this committee who decide that we need to inform everybody, all the doctors who are the prescriber, that hey doctor, look, this is the new problem, this is the new signal which we have come across about our molecule. It will take little time for us to include this in our label, but before that we are just giving you this particular communication. So there where the dear healthcare professional or dear doctor letter comes into this, then you need to have legal, this legal, I will come to you little later, and you will realize that how important it is to have a legal team in a corporate sector, especially to help the pharmacovigilance people. And then, of course, you have training and development. You have to continue to train your pharmacovigilance people, continue to train your stakeholders, including your sales and marketing people. Can we go to the next slide, please? Is it, Krishna, I would like to know whether this is making sense or this is too preliminary, and whether people know about this.
No, sir, this is what exactly we required from your experience.
Okay, good. So I'm happy. So friends, this is a simple flow sheet which actually explains the entire chain, what I was talking about. So we have the first, you can see that you have data. And once you have the data, naturally, you are getting so many cases that manually you cannot do data analysis. This data evaluation is being done by database. I'm sure you must have heard about ARIS-G, you must have heard about Argus, you must have heard about PV24/7. So there are a lot of other new databases are coming, because humanly it is impossible to evaluate thousands of the cases which are coming to you, and you cannot have an Excel sheet and do all the analytics using Excel sheet. So using this database, you do a lot of analytics, and once this analytics are available, it will also have signal detection, and based on this signal detection and evaluation of benefit risk analysis, which I have been explaining, you will have some signals or some communication which will go to all the stakeholders. So this communication will go to the regulatory authorities, this communication will go to the doctors, it will go to the patient through PIAL, and it can also go through in form of the dear doctor letter or dear healthcare professional letter, so that people are very well informed. So let me explain you one particular example of the communication. I'm not sure whether you know a black box warning. Have you heard of black box warning? So a black box warning, when the FDA says that, dear company, we would like that your label, your PI, the USPI should have a black box warning for this particular product. So you would, when you open the USPI, the first thing which will come in front of your eyes is a very thick black bordered box in which there is a chetawni, where there is a warning to anybody who is taking that drug, that by our experience we know that this drug can cause cardiovascular event, like what happened with your the very famous GSK molecule, which was used as anti-diabetic, and they used to have this black box warning. But then when they realized, the company realized, and FDA realized that this is becoming very dangerous, this particular molecule was withdrawn from the market. Okay, so this black box warning, when you put in the form of the PI or USPI, what you are actually telling the patient and the doctor, that we have given you information about the risk associated with this particular drug. So now it is up to you. You have to take an informed decision whether you should continue with this drug or whether you should not continue with this drug. So now the ball is in the patient and the doctor's court, that he has been informed by the marketing authorization holder about the possible risk with this particular drug, and then they have to take an informed decision, like you take an informed consent form while doing a clinical trial. It is something like that, but a very lighter and dilute version of this. Can we go to the next slide? Yes. So this will give you today's, PV domain in the industry. This is very important for all budding professionals, my students who are doing B Farm, those who are doing M Farm, and those who would like to make pharma industry as their career path. So there's a remarkable growth, and has been acknowledged uh, that India is a competent global player. And that's why, as I was telling you in my previous slide, that most of the MNCs have given their medical writing work, like writing of RMPs, writing of PSURs and PEBRAs, and also doing ICSR processing to India. Okay, so what are the achievements? So, right from case triaging, I don't have time to explain all these terms. Just understand that right from the first step, when you receive the case, there the triaging start to submission of the case to the regulator. This is the entire end-to-end case processing, and including medical review and signal management. We are doing annually 3 million cases. Can you believe this? 30 lakh cases are being processed in India. This makes 80% of ICSR processing of the world. Global 80% of the cases are being processed in India by all these big KPOs like Cognizant, TCS, Accenture, Covance, and so on and so forth. This is my last year's figure, I could not update this. We have around 6,000 FTEs, employees in these industries who are continuously working for pharmacovigilance. 30% of aggregate reports are being written in India, and 10% of RMPs of the world are being written by India. So by this particular data, what I'm trying to explain it to you that there is a very big demand for pharmacy graduates and postgraduates and doctors, whether they are BAMS, BHMS, or BUMS, or they are MBBS, or whether they are MDs. There is a big demand for these doctors to be part of the PV processing, PV juggernaut of the industry, because doctors are considered two level higher up than pharmacy graduates, because they are directly trained into medical assessment of the cases, medical assessment and medical writing. So they start at slightly higher level of the PV chain in the industry. There are certain challenges and learning, we had to create a big pool of PV professionals who are trained, learning in terms of enhancing productivity and quality. And today we have lack of trained manpower. So what I mean by lack of trained manpower, if some company comes to Nipur and picks up maybe 20, 25 pharmacy graduates to be part of their, KPO or case processing unit. So suppose if they pick up somebody in month of October, they do not become productive for 3 months. So there is almost a 3 months gestation period before they are being trained with entire nuances of pharmacovigilance. So they have a boot camp training, then they do what is known as a training environment of database. They are supposed to do some trial cases. So each KPO has got various limits that you have to do 50 number of cases correctly, 100% correctly. Then only they certify you to work in the production environment. They will allow you to get into production environment of any pharma company where you would start analyzing cases on your own. So what I'm trying to say that currently the KPOs are struggling to have trained manpower, whereby they don't lose the 3 months of time after hiring these people, and these people are already trained, and with little bit of more training on databases, it can be RSG, it can be Argus, it can be 24/7, or it could be Vigibase or Vigiflow, and people can start working directly into the production environment in their company. So this is some of the challenges which we have today. Next slide, please. So besides this, as I was telling you, just imagine now if J&J or Sanofi is working in 100 countries, how do they control those countries? So they have a local operating, company affiliate office where they will have what is known as local safety officer, they call local safety pharmacovigilance responsible person. There are various designations which are used. So you can be looking up to, for Indian company, for foreign companies who have their offices in India, you can be looking up for a local operating company's pharmacovigilance officer position. Now, today, according to our new regulations which have come, the guidelines by by CDSCO, which was put into effect from Jan 2018, we are supposed to strengthen our existing PV setup. So according to that, you have to have one person which is known as pharmacovigilance officer in charge. So he needs to be a pharmacy graduate or a doctor. So you can be looking up for, because now all Indian pharma companies are supposed to have a pharmacovigilance function. And these are some of the requirements. So they must have a PVOI, they must have a master, what is known as PSMF. So here we have differently named it, but then we need to have a document which is known as the master file for pharmacovigilance, where you will include all the provisions, what you have, the infrastructure, the training, the database, how many people you have, what are their reporting lineups, what are their JDs, everything is included in the PSMF. So now Indian companies also will be forced to enroll or employ pharmacy graduates. So I also see that you have more employment opportunities coming up, because this is a mandate. Of course, currently this is just a guideline, but as I was told by some CDSCO officers and some PVPI people, that this is going to be made mandatory, because we are expecting inspections from the CDSCO people as well. So, but the biggest problem with our Indian companies, as I have put it in learning challenges, it is financial constraint and cost center. So, whenever, if you want to have a big database like ArisG or Argus, for only the first year itself, you will have to put in something like 35 to 40 lakh of rupees per year to have this safety database to use. So this becomes a big financial constraint for Indian companies. And since this is not a profit-making business, this become a cost center, and they would be very, very hesitant to put so much of money, invest so much of money. Can we go to the next slide quickly so that I can complete? Forget this international scenario. Let us go ahead. Just go to the next slide. I can. This is a pharmacovigilance in India. As you know, that we have now new pharmacovigilance program, which was effective 2010, and we have got more than, I think, 300 peripheral centers, where again you have employment opportunities like coordinator, center in charge, and there are so many other things. I think there are unit of 4 to 6 people depending how much of work you get there. So that is one. And so we are getting, but unfortunately, I was just seeing the latest September newsletter. We are still getting something like 6,000 to 7,000 ICSR at the national level. So that is very less number of ICSR being reported. Our reporting rates are very, very low worldwide. So we are less than 100 rank, I mean below 100 rank in reporting incident of ADR, and we have to create awareness among our healthcare professionals and ourselves. When you become a healthcare professional, we have to ensure that more and more ADRs are reported. I'm not asking you to generate fake reports. What I'm trying to tell you, that when you come to know, we have to create this awareness. When there is an ADR, this needs to be reported, and it is reported in time to PVPI and also to the marketing authorization holder. Next slide, please. Next slide, please. So this is an old slide. So you can imagine these are the places where you have pharmacovigilance program working by UMC. Okay, these are the member countries. There are more than 130 member countries, which might have gone to increase. So I always show this to my budding professionals. So today you are sitting in Chandigarh. You can see that you cannot see Chandigarh. You can see definitely India. But if you have sufficient experience in pharmacovigilance, you can be seeing yourself sitting in North America, Latin America. You can see yourself sitting in EU or in Australia or any other country where UMC is operating and which is part of the UMC or WHO pharmacovigilance program. So this is a big, huge opportunity of pharmacovigilance outside India as well. That's what I'm trying to explain it to you. Next slide, please. What is most important, which I am trying to say, that this is PV is a serious business from a humane angle and also from a commercial perspective. So that's what I'm going to prove it to you. Just go to my next slide. That you know this. You must have read that it is almost 1 lakh deaths taking place every year because of adverse drug reaction. 35% of the hospitalized patients experience an ADR. Next slide, please. ADR, or 2 million serious ADRs per year. 1 lakh deaths nearly. ADR is the fourth leading cause of death ahead of pulmonary disease, diabetes, and AIDS. So you can imagine the extent of drug-related problem which is existing in the society. So by creating proper awareness or taking care of the patient properly as a healthcare professionals, there are reports in the publications that 50% of ADRs can be eliminated or minimized. So you can imagine if 1 lakh people are dying, if you contribute to 10% reduction, you would be saving at least 100 people every year because of your good work as a healthcare professional. So this is the human face of pharmacovigilance. Let us go and see the financial face. Now, all the companies who are putting, go to the next slide, please. All the companies who are putting in millions and millions of dollars because they know that if they fail in maintaining the robust pharmacovigilance system, they will have to pay through their noses. And these are some of the figures which I have put in front of you. How Wyeth has paid for Fen-Phen, how Bayer has paid for Becol. The latest is Merck's Vioxx case. They have put aside $50 billion for all the cases which have been filed against them for CVS adverse drug reaction caused by Vioxx worldwide. The first case which was gone in favor of the patient who said that she lost her husband because of cardiac arrest, and he was a regular Vioxx, consumer, and the jury said that she is justified, and juries asked Merck to pay $350 million to this particular lady, and she got a reimbursement of $350 million. Okay, because her husband died because of using Vioxx and having cardiac failure. So dear friends, no company can afford this type of legal battles and legal fines, and that's why company is investing billions and billions of dollars in having a very robust safety system so that they can have signals, they can inform the people in advance, they can withdraw the drug at the first phase, second phase, third phase, or even immediately after introducing the drug into the market. There are there are almost 10% of new molecules being withdrawn every first year of the introduction because of the safety reasons, because they are getting a lot of safety issues after introduction into the market. Next slide, please. So you can imagine also, this is my last slide, the share value of Merck was when they took this decision of removing Vioxx from market was near 45 $40, and overnight it plunged to around $25. And they had, according to market, this thing, they lost $27 billion in market capital. So these type of financial losses, no pharma company can really afford. So this was all I wanted to talk to you. In case if you have any, I really hope, I'm not sure whether this was really a useful presentation, whether you knew everything before what I talked to you about, Krishna. I don't know whether I came up to your expectations, and the students who are hearing me, they really got something to take home. And I will be happy if you can give me some feedback, and in case if there is some question quickly, I can take up, because
Sure,
we
sir.
are already 10 minutes late. Yeah.
So certainly it's a very useful presentation. Sir, before that, can I have your video, please? I think people want to see you.
People want to see me. I do not have Shah Rukh Khan's face, so, but
But still,
not worth looking. No, somehow I'm unable to open my video.
okay,
So it's okay. It's okay. They can go to my LinkedIn profile, and in my LinkedIn profile, my photograph is there.
Sure, sir. Thank you. So thank you for bringing all your experience in pharmacovigilance. And no, that gives a very good information to the audience. And especially, I think by this time they understood that how much important the pharmacovigilance also in healthcare sector. Sir, if possible for you, we can take few quick questions.
Yeah, sure.
Especially, I think the questions from Varsha Ways, as she is very active. She is attending all the sessions and keep posting the questions. And her main query is, who can exactly do the causality assessment? Is that only MBBS people are eligible, or anyone can do the causality assessment?
Okay, so there are a lot of algorithms available now, causality assessment, as people must be knowing. We have WHO assessment, then we have what is known as decision trees made by the pharmacovigilance department of a lot of pharma companies. They normally depend on medical graduates to do the causality assessment, because as you know that there are a lot of therapy related or molecule related questions which needs to be ruled out before you confirm the causality with a particular molecule. So I mean, even if you want to decide on the timeline relationship with the administration of the drug and when the ADR was manifested, you must know about the half-life of the particular molecule. So this, we normally don't have this type of information available, or we don't expect the pharmacy graduates to know. So most probably, I have seen, according to my own experience, the doctors, which we know as drug safety physician, they are the people who at the end of the case processing ladder, they are supposed to do the seriousness assessment, they are supposed to do the causality assessment, and they also do the labeling assessment. These three assessment is normally done by doctors. In case if you have a pharmacy graduate who is by experience now very mature, and he has also picked up a lot of medical knowledge and all that, so if you are sure, then you can use him. But by and large, I think 90%, 90 to 95% of causality assessment is being done by doctors in pharma industry.
And she is having one more question, sir. Actually, even we have answered this yesterday. I think she is still want to hear from you. If any, especially the medical device named as a drug, and when you get any adverse reaction for that, adverse event for that, we need to report as adverse drug reaction, or we need to report for material vigilance.
I think material vigilance and PvPI is in conjunction, like in US FDA or even in EU. In fact, in US FDA, the form itself is a combined form for ADR and for drug drug device. So this is a typically drug device form or case which is being reported, and then both the department has to take cognizance. It is like, if you have a product quality complaint, okay, and which is associated with an ADR, so this may go to a quality designee as a product quality complaint, but then he has to share this particular case with his pharmacovigilance colleague, so that they also take care of the ADR which is associated with this particular case. And that's why they have to also do a monthly reconciliation so that they don't miss out anything, whether it is a PQC or whether it is an ADR. So even the material vigilance and the PvPI, they must be doing a reconciliation among themselves so that they have a case registered as a medical device case, and they have also a case registered for the drug which is being used in the form of the device.
Yes, sir. Thank you very much, sir. Sir, there is one request from my side. Most of the participants, we are getting very good feedback, and all are saying that it's a very informative session. And my request is, sir, all the students who joined here, they're very young graduates of different courses. I think most of them want to opt pharmacovigilance as their career in future.
Yeah.
So what is your suggestion, sir, from your side, being an industry person from long time in pharmacovigilance? So what kind of skill set they need to have before approaching any industry for their job?
So you can share my email ID with them. Very simple, moin@pvcon.in. I am also on LinkedIn, so they can definitely join my LinkedIn. There are some training courses going around, which they can think of, so that what I was talking to you and the people, that if they go with this particular training program already done, probably they will have better chances, because they would know the nuances of pharmacovigilance, the entire chain which we were talking about. And I found that some knowledge of the databases and all that is always very handy. So some IT related skills and good English and Medra coding also becomes a very important thing. So MSSO offers Medra coding courses, online Medra coding courses. If they can do that, this will definitely help them in getting a better leverage than other people who have not done. So IT skill, good English skill, Medra coding, and having some training programs which is there available online. So there are a lot of companies who are offering online courses besides we offering this online courses. So I think this could be a useful thing for them to begin with. And I will be more than happy to answer anybody who comes back to me through email or my LinkedIn. I'm very busy. I'm traveling most of the time, but then I always try to help youngsters who would like to come up in the industry. And I can tell them, one of my student, while talking to them like this only, I had used the same slide and I said, look, today you are in India, tomorrow you can be sitting in EU as a QPPV. And one of my student today is a QPPV and he is sitting in Amsterdam and is, working as a QPPV of a big multinational company. So this can happen to anybody who is currently looking at. So you have to be ambitious and you have to work hard and you have to have commitment.
Thank you, sir. Sir, thank you for raising that the last word, QPV. I feel it's a very important in terms of pharmacovigilance. Can you just maybe just in 1 minute, can you say how much important it is? I think for the audience.
Dear friends, QPPV is the most important single person in EU pharmacovigilance setup. So without having a QPPV, you cannot have a pharmacovigilance, you cannot have a PSMF, or you cannot register yourself as a pharma company. So if you want to register yourself as a pharma company in EU, you need to show them who is your QPPV and who is your backup QPPV. You don't have to have only QPPV. You also have to have a backup QPPV who will work as a QPPV, and he is the sole person who is responsible for the safety of all the drugs of that particular company. So in case if EU or EC has got any problem, about any drug of that company, they will not go to the president or CEO of that company or medical director of that company. They will directly call up the QPPV. So he is supposed to be available 24 by 7 to European board. Okay. And his mobile number, his landline number, and his email ID is all known and registered in Udra Vigilance and available to the entire 28 member state besides the EB. Okay. So that important it is. And they are paid handsomely. I mean, they are just paid handsomely. There are also known as contractual QPPV. So a smaller company who cannot have QPPV on board, they can approach to Dr. Krishna. Dr. Krishna, how about you becoming QPPV for us? And all such contractual QPPV are being paid something like 1,000 pound per hour of working. So it is almost 1 lakh rupees per hour of QPPV services. But
Yes,
that
yeah,
you need to have that sort of skill set and knowledge.
yeah, friends. So that much powerful position it is QPPV.
Yes, yes.
So hopefully, maybe some of the audience who are joined here, sometime maybe you will be having that kind of position. So please let us know when you go to that position. And I'm sure that
I will
what
be, I will be very happy to know that somebody has taken this inspiration from me and aspired to become a QPP.
a prestigious position in pharmacovigilance in European Union.
Yes.
So thank you very much, sir, for joining with us and sharing all your knowledge. I'm sure it's going to be very useful for the audience and shown they're going to be very good professionals in terms of pharmacovigilance. Thank you once again, sir, from NIPER side. Thank you for all your support extended. And one last sentence from my side, as yesterday Mr. Stenolson mentioned,
Yeah.
recently the ISOP, that International Society of Pharmacovigilance, announced a South Asian chapter,
Yeah.
and Moindon identified as one of the leaders for that. So maybe we can soon see some more activities under the chapter, so that the concept of pharmacovigilance spread across these Asian countries. Thank you very much,
Sure.
sir. For
Krishna, I assure you that as soon as things are normalized and I can travel to Chandigarh, I'll be first person to coming to Naipur. Pramil also has been inviting me for very, very long time. He's a very good friend of me, Dr. Pramil Tiwari. Okay.
yes, sir.
So
Yes, sir.
we can have a face-to-face workshop for all the students of Naipur.
Sure, sir.
Okay.
Sure. That will be
Take
glad to
care.
have. Thank
All
you
the
so much.
best, everybody. All the best. And please do write to me about your feedback, your questions. I will be happy to understand that what I presented was really useful to you. So even if 1 or 2% can write to me that this was useful, it will be very satisfactory to me. All right?
Thank you.
Take
Thank
care.
you very
Take
much,
care.
sir.
Bye-bye. Have a nice day.
Thank you, sir. Bye, sir. So, have participants, we are at now 11:22. So we'll take a quick break and then come back faster, 11:30, 11:30, because we have already our next speaker who is going to be show you the demonstrate you various databases they are using in industry. That's going to be more interesting, like the first session where the demonstration happened. So come back faster. By 11:30, we can start the session. Thank you. Welcome back, everyone. We are come to the last session of this National Online Workshop on Pharmacovigilance. And it's going to be a very interesting session, the last one, where we have one more experienced person from industry, Dr. Amrit Raja, the Senior Director for Product Management in Vitrana, Bangalore. And if you see his biodata, Dr. Amrit is a physician and holds masters in clinical pharmacology with hands-on experience in human trials, the pharmacovigilance, regulatory affairs, and comes with over 18 years of experience in healthcare, IT, life sciences, and also the pharma. So it's a very huge profile the SAR is having already. In the last decade, he has held strategic leadership roles in industry that is leading organizations such as Novartis, Oracle, IQVIA, Covance, and has been instrumental in driving process, productivity, and automation initiatives while identifying challenges and creative, innovative solutions. So once again, we have a very eminent speaker from industry who is going to be explained to us some of the databases commonly using in industry in terms of pharmacovigilance. So with this brief introduction, now I would like to welcome Dr. Amarjit for explaining the concepts. Dr. Amarjit, now the screen is yours.
Thank you, Dr. Krishna. It's been a pleasure to interact with you. I hope I am audible.
You're audible. Can we have your video also, if possible?
Oh, yeah, sure. Am I visible now?
Yes, we can see you. So participants will be happy to see you. That's why. Yeah.
My pleasure. My pleasure. All right. So thank you for that introduction. NIPER has been a very premier organization. Congratulations for being a part of NIPER. I've been associated with a number of talks in multiple different forums, but I feel equal privileged to be associated with something that's happening within India, something where we have our own people coming and talking about what you've done. Just like we have Microsoft, Google, the prime people who are transforming the technology space. It's all Indians, right? So we have majority of them Indians. So, it feels very proud that even in the technology space for PV, right, it is us who are kind of dominating that particular spot. So with that in mind, right, so let's quickly jump onto the topic. And I will go ahead and start sharing my screen. I hope you can see my screen.
Yes, we can.
Right. Okay. So the topic that was given to me was around drug safety technologies, and it was actually drug safety databases used in the industry and a demo on ARC safety. Right. But I would like to take one step back and talk about drug safety technologies, because database is just probably 10% of the entire range of technologies that we basically operate to keep this world going for the drug safety team, right? And thank you for that quick introduction, Dr. Krishna. So we'll kind of cover this time looking at briefly around history. I know that most of you have become historians by now, right? Because you've heard PV going back and forth. So a quick recap and how technology sits in there. A bit of an introduction to what we're trying to achieve, right? What kind of challenges that we face in PV and how technology has come to that rescue, right? Then we look at who are the major players in this particular space. Then we will jump into the demo, right? And then we will look into what are we trying to basically automate and where are we moving through this technology, and what is the path forward, right? What is the life looking like for technology, right? So I hope this is the comprehensive range of topics that we are trying to cover around here, right? Just a quick recap on where we stand. Almost all of us have known about the thalidomide tragedy. We start from 1961 from thalidomide tragedy, but then rolling back to 1848, where we had one of the patient, Hannah, whose death was caused by chloroform. At that point of time, we did not have enough tools, measures, and thoughts around what really led to Hannah's death after she had actually, was she was just given chloroform for removal of an infected toenail, right? Once she was given a chloroform, she took her last breath, and nobody knew why she died, right? So eventually, once that formal record of somebody where we had some doctor reporting that Hannah had been given had was suffering from a toenail infection, was given chloroform, and during the procedure she passed away, right? So this document was relevant in many different forums, right? But for PV, it became relevant primarily because we had the four identifiable criteria: a reporter, a patient, a drug, and an event, right? So that kind of traced back to 1848. So we are like looking at 150 years, 150 plus years, or 160 plus years of history that we are trying to trace back, right? And then we had many different milestones that we could dig out from history, right? And then probably 1995, once we had the EMA set up, and then 1968, we had WHO program for drug monitoring. I still remember my days when I had joined Novartis. At that point in time, we still had so much of paper, so much of reports, so much of things coming in, right? It was not just limited to Europe, it was not just limited to US, but then we wanted to look at all the parts of the world on how we can capture this information and put this through. More than understanding that this brings in product um, safety, right? It also, like in previous talk, you heard Mr. Mindon talk about the amount of money that is at stake, right? So it was critical for all of us to bring this domain, pharmacovigilance, to be a very serious domain, right? And while we are trying to look at cost saving, we are trying to look at globalization, we're trying to bring in as much information as possible. We're trying to capture information, we're trying to put them into a repository, and then we are looking at analysis, and then we're looking at creating reports out of them, and then submitting it to the regulatory authorities, right? The number of pharmaceutical companies that are existing in the world, right? So everyone are obliged to be responsible for their own product. Imagine a world where you had no computers. Imagine a world you had no internet, where you didn't have, applications or softwares that can process this information. So you would be moving huge amounts of paper from one side of the world to the other side of the world, right? And then huge amount of manpower to read through all those documents, right? So it is inevitable. What I'm trying to tell you here is it is inevitable to embrace technology. And PV is one such science where we can say that it has matured over the last 173 plus years. It's matured in a way that we are able to understand what we're doing, right? But although we say it is matured, but it has an age and it's still maturing, it's still refining, we are still, bringing in new regulatory changes. We're trying to evolve our understanding of pharmacovigilance, right? So as as that, we are in 2021 and 2022, we are still waiting for an E2BR3 format implementation timelines, right? So, and then we have 2024, we have IDMP, we have a certain pipeline that basically has been sitting with us, right, from a history perspective. So, what is a pharmacovigilance safety database, right? So it is a central repository for all your individual case safety reports, or we call it as ICSRs. They are collected for a company's medicinal product from all the sources globally, right? So it could be a database, could be anything, electronic or physical, whatever. But now we are talking about mostly electronic. Why do we need a technology in PV? Okay, so I basically broke it down into 4 different reasons, and the reasons are evolving as well, right? At this point of time, what we know, we're trying to assimilate. So there's increasing use of medications via access to healthcare. So we know that more and more population across the world are gaining access to healthcare. And as they gain access to healthcare, there are more prescriptions, there are more people consuming medications, right? So, in a study by IQVIA Market, they say that the annual growth rate is at around 3%. We're talking about something like $750 billion of money that are spent year on year on use of medications, which was probably a half 7 years back. It's now kind of doubled, right? So as we know that more and more exposure to medication, more population exposure to more medications, I think that's a combination that will result in us understanding that we need to capture more and more data that kind of generates out of it. People also getting increased awareness about reporting, right? So people now started knowing. I think the institutes like PI and WHOs and the organizations, the MHS, they're all promoting that if you have any side effects, please report it. Most classical example is when you had a COVID outbreak, right? And you're taking a COVID vaccination now. I think they've been reporting saying that if you have any adverse event, please report it onto a portal, right? So increased awareness about the reporting is also one reasons why we are getting more and more adverse event reports being databased, right? Pharmaceutical manufacturers, one is although there is a regulatory compulsion for them to report, but their own interest and in order to understand how to improve the performance of the medicinal products, right? And and we know that as we are moving forward, there's also increased cost in conducting vigilance activities, right? With so many number of reports that are coming through, what are we going to do with that? How do we move forward from there, right? More and more channels of adverse events getting reported. Regulatory reasons. So there are evolving regulatory guidelines, right? So you have combination medication coming up. So the guidelines kind of talk about how do you capture combination adverse events, summarizing and crystallizing safety events for submissions in PADER, PBERS, and your DSURs, right? And then you also have all these kind of compiling towards requirement for periodic reporting, and then signal management, and then benefit risk dossiers that needs to be submitted. Technological reasons. We know that we have evolving data collection methodologies, right? Things like, you have your variables, you have your Fitbits, you have evolving tools that allow you to capture more and more data, right? What are we going to do with that set of data? It's something that we need to really understand. How much of that can be related to the adverse event? Can we capture? So you know that the latest Fitbit Ultra is able to conduct ECGs on your phone, right? And based on that ECG, you can even detect that you have an abnormal heart rhythm, right? And you know that there are so many drugs that causes abnormal heart rhythm, right? So can we use these sort of tools to start capturing adverse event information, right? And the amount of data that this sort of wearable devices pushes on, right? So you need a huge amount of you need a different technology called as big data analytics, right? For Hadoop and other databases which we have used to kind of come up with some meaningful insights out of them, right? And then there are growing need for automation. So everywhere we are looking at automations, we are looking at robotic process automation, we are looking at natural language processing, we are looking at artificial intelligence, right? How can we make most of the work that is more logic based, something which is more driven, something more monotonous, right? Earlier, I remember I started off my career by looking at adverse event and then trying to find out if this adverse event is serious or non-serious, and then taking a color-coded file, right, and then putting it into it, and then looking at, okay, so this is the death case, this is in a red file, and people have to move this in the red file, right? So now technology takes care of it. It identifies, it puts a flag that this is a death case. It puts priority numbers. It presents to the user as to, this is something that needs to go out really quick. And if it is not, it'll have a clock ticker that says, okay, so now this is your countdown time starts, right? So we're using technology to move things forward. So when we have technological growth happening, right, versus we have a, a an area that requires that bringing them together becomes a very critical responsibility for us to ease do a lot more than what you're currently doing, right? So I was talking about the challenges that PV technologies are able to address, right? So adverse event volumes are growing with increasing disease complexity, right? Not only that, we are also looking at newer pharmacovigilance for orphan drugs, right? The drugs that are not very commonly used, but we're using them for activities, right? So we're talking about wearables, social media, right? Social media driving real-time data, right? So even how are we capturing adverse events that are reported on social media, right? An Instagram post or a Facebook page, right? So all these areas become source. You cannot ignore, right? You'll have to go back, understand, mine out that information, talk to the patient, and then try to make sure that or validate that it has the right safety information, bring them on board. Global regulations are becoming more and more complex. You'd have heard about what is called as PII or patient identity identifiers, looking at global data protection policies that have come in. Countries are trying to protect the data not going out of their country to and expose what's happening within their country. A very typical example is how China even prevents any body samples moved out of the country, like blood. You cannot move blood. You cannot draw blood from Chinese national and send it to Singapore for doing the tests, right? So you need to have all samples of information belonging to that particular country within the same country, right? So the regulations are becoming more and more complex and they're more dynamic. Another very good example is a Brexit, right? How Brexit political situation impacting the global regulations, impacting the regulations that are more local to Britain, and how they kind of moved out of the EU, EMA, and then have their own regulations. And in the process, we have a new country called as Northern Ireland being set up, right? Northern Ireland has to report cases to us. So a political regulatory situation that is dynamic. How is our technology able to manage in that kind of an environment, right? Product portfolios and therapeutic areas are getting increasingly complex. 10 years back, we never heard of something called as orphan drug research, right? Or orphan drug trials. So now those are becoming more and more explicit, right? Earlier we were talking about lifestyle modifications, more the anti-diabetic drugs, antihypertensives. Then came an era of oncology and cancer, and that kind of dominated almost a decade, right? And now we are looking at personalized medicines, orphan drugs. We're looking at rare disorders like musculoskeletal disorders and things like that. So in that situation, we are looking at different sort of a safety profile, a different speciality within a therapeutic area that we're really looking at, right? Then raising organizational pressures to minimize cost, right? So because we are not getting a revenue out of PV, right? So so you are doing PV to save the drug from getting into investigation or preventing them from getting banned, right? They are not the money generators. Most money generating arm of the pharma organization is the sales arm. The more they do sales, the more they invest in sales, the more revenue comes back to the organization, right? But PV is more like a salvage organization, right? So you're trying to have PV cost reduced in the recent years. So you're moving a lot of jobs out of what we're paying for simple manual work. We're bringing in robots, we're bringing in automation, trying to minimize the cost over there, or move the work to low cost regions like Manila, India, and things like that, where we're able to do the same work at a much lesser cost from, and you're able to save cost from a labor arbitrage, right? We also look at data sciences, automation, evolving, where we harness these technologies for doing signal detection, for getting your accuracy to have that product event relationship scrubbed up and clean, right? Automation involved in case management, right? How can we reduce the total time spent on a particular case, where we talk about efficiencies that needs to be built within the organization, right? Also focusing on the risk benefit analysis and signal management, that is the need for advanced safety data analytics tools. Here mostly we look at what kind of models and algorithms kind of come into place that allows us to measure the delta between what was the safety profile a few years back to where we are standing today, and how we are improving on them and how we want to move forward. And the last one is consumerism is demanding increased focus on patient centricity, right? Whatever work you do at the end, if it doesn't get translated to the patient, right, in making sure that the drugs that are available for human consumption is absolutely safe, right, this work will continue to go. And our job in terms of harnessing technology, the purpose is to ensure that the drugs are made more and more safe as they kind of come in. New drugs coming in, as they come in on board, they need to be safe enough for human consumption, right? So these are the constant challenges that we encounter while we try to harness the technologies in managing multiple different challenges within the pharmacovigilance domain. So I'll just pause here and check from the audience. Do they have any questions till now before I move forward into the main aspects of the technology?
I think at present they don't have any questions. Maybe we can move forward and we can take up at last if they have any.
Lovely, lovely. Thank you so much. So when we talk about pharmacovigilance, safety database, and technology, right? So this is a comparison of where we stand from a PV process to what we talk about technology, right? So where all have we harnessed technology? If you see on the upper grid, you could see activities like pre-intake. Pre-intake will include things like, literature screening, literature curation, right? And things like that. Or say, for example, connecting with a clinical database. So those are all pre-intake. Intake is when you have a definitive adverse event form that gets filled in and sent to the case processing group, right? So in intake, they would identify if you have all the criteria that are required for it to be a valid ICSR, right? And from there, you move it to case processing, which will be more elaborate information where you're able to fill in as much details as possible. It could be related to the patient, to the product, to the event, perform certain analysis, write up a narrative, and also, attach the source of your information that has come through, which qualifies it to be ready for ICSR submission to regulatory authorities, right? And depending upon the type of case, depending upon your process, you will also have a medical review, which will basically talk about causality determination, will talk about temporal relationships, they'll talk about or validate your re-challenge results, which in turn talks about your causality, right? Then you also have, you know, analytics conducted on the final document. And after you conduct your medical view, then it gets into submission. Most of you might be wondering, where is the quality check? Okay, the quality check is a part of the case processing. For all practical purpose, we will consider it to be a part of case processing. Okay, but these are essential elements that are critical for us to make sure that the individual case information gets submitted to the regulatory authorities, right? Submission. So while we run analytics on a fully formed case, we are able to generate aggregate reports like we talked about PSUR, DSUR, and papers and things like that, right? So that gets into submission. And also, you will have signal management being done both from periodic reports as well as ICSRs, right? And from signal management, you have BRMs written, right? Benefit risk management plans written and submitted to the regulatory authorities, right? So the ICSRs, aggregate reports, as well as BRMs all become our obligation to the regulatory authorities to ensure that to give the regulatory authorities the confidence that the product is absolutely safe for human consumption. While you're doing this mainstream job, you also have a certain set of people or a process that oversees is the process going in the right direction, right? Also takes care of quality management, and based on that, whatever findings kind of come out, we'll also have to manage kappa, right? Kappa is corrective action, preventive action, and all these become a very integral part of your safety process. What you've not talked about is safety technology team, right? And within the technology team, you have development teams and you also have support teams, right? So what technologies kind of complement the process above is mentioned in those, dark blocks. So you have a mail intake, then you have literature curation and processing, right? Which extends across pre-intake to intake. Then you have case intake, where you get cases from two different partners, right? So you have two pharma companies who are partners with each other, right? And they would want to exchange the information, like, you take my case and report it to your regulatory authorities because you have my product marketing in your country, right? And therefore, they would have to get some sign-offs there. Then you have the traditional case processing applications. So we will talk about one such a traditional case processing application is Argus Safety or RSG, where you conduct your or you conduct your intake, case processing, medical review, and submissions, right? Then you have dedicated submission portals, right? If you're able to do your case processing, medical review, you have submission portals, which allows you to do specialized submissions, right? Not only to regulatory authorities, you'll also have to do submissions to ethics committee, right? You'll also have to do submissions to the principal investigator who are a part of the trial being conducted in a multicentric trials, right? So this sort of a distribution that happens not only to regulatory authorities, but also related parties. You have specialized applications or technologies that support that, right? Obviously, from an analytics standpoint, we have analytics and reporting applications, very old ones like Power BI, Cognos, and Analytica, Informaticas, and things like that. So for aggregate reporting, you have document authoring. So if you had a session on aggregate reporting, you'll know that you're getting information for a certain product over a period of a certain duration, and you're trying to summarize that information, and then you're kind of creating some analysis tables. You're creating case data analytics. You're creating summary tables of, like, say, for example, the liver toxicity in this particular set of patients who are taking this drug, or for that matter, any such information that we want the regulators to be made aware of, right, will be a part of your document authoring tool, right? And then you have the signaling app. Signaling app basically is something that allows you to bring together database information for your company, right? And then they will look at the database or the information that's available in the public domain. When I say public domain, I would say, morely to what is sitting in the regulatory authorities, right? So the FDA, the EMA, and say the PMDA or the MHRA, and then you're trying to bring all that information together and then comparing what is the power of your or the significance of your product event relationship to what is existing in the background. So there are a lot of tools that require you to do those statistical analysis and statistical attribution that can determine a certain amount of relationship of the product to the event that is in question, right? So you have signaling app that has diverse functions. It allows you to list out all the items in rows and columns, or it gives you a very visual chart in terms of understanding what we call as a heat chart, which says that, okay, so this is the most commonly occurring side effect for this particular product. So it will be a chart of all your events, but with various degrees of, redness, what we call as heat chart. And it will give you a very clear understanding to say, okay, so this list of unlisted adverse events are something that we need to be careful of, right? And then they kind of manage the signals that kind of come out of it. Then you also have a quality management application that allows you to manage all the quality issues that kind of come either from product quality complaint or from the quality of the ICSR that you process, to managing audits and inspections, right? And finally, you have Kappa apps that allow you to document, monitor, and fulfill your corrective and preventive actions. Right? So these are a wide range of technological application, technological support that a PV is actually erected on, right? So if you have a flag of PV flying, then you have this pole that actually holds that, and these are the different foundation elements that actually makes the entire PV process very, very strong. And what we're talking about in today's session is on traditional case processing application, right? So now you get an idea about why I kind of moved it from safety database to technology, right? So what we're seeing is just what I would say a backbone, but then there are other critical elements within the organization called as, or an organism called as a pharmacovigilance, right? So this is the landscape or a technology landscape in which we kind of operate. So when we look at who are major players in technology, suddenly it gets very colorful, right? Like my slide here. So when you see, this is again the top 10 list of people, or top 12 or top 15, whatever you want to call it, who have been operating in this particular space. Oldest being Arcus Safety, Oracle Arcus Safety, followed by Aris Global. So Arcus Safety is in the market for probably now 20 plus years, right? So 1995, 1991, 92 onwards, right? It's been here till 2001. So close to 20 years they're kind of touching. Aris Global, very similar age group, right? And then we have the new generation tools that have taken over a lot of these, players, the older players in the market. The reason why they still remain to be dominant is because they form the backbone that are supporting the top 10 highly earning pharmaceutical organizations, may it be J&J, Novartis, Sandoz, GSK, AstraZeneca, Pfizer, Merck's, right? So they're all around the first two tools basically support them, right? From being a part of the backbone of your PV structure. Whereas we have other organizations like Sophos who have a very advanced adverse event intake. So they have, you pass in any email that has any adverse event, it kind of extracts this whole information and loads it into Argus Safety, right? Absolutely not requiring any human intervention. Then you have vigilance, which is a real-time surveillance. It keeps running its scanner across the different pharmaceutical as well as the regulatory authorities and tries to point out, and anybody who subscribed to that gets to understand what is that area that we need to focus on. Vitrana, from where I come from, focuses heavily on a platform called as Highlight, and they have around 35 different products that kind of fit anywhere. What we call as accelerators, right? PV accelerators, which means that they accelerate your process. So they improve your efficiencies by 15, 20 times, and enhances your data quality by some 70, 80 times, right? Making sure that there are absolutely nil human errors and things like that, right? So then we have PVNet, which has again another suite of products, Genpact. They have come up with Cora Pharmacovigilance, which again, is another alternative to target safety. Then we have Viva Vault. They look very beautiful in terms of how they are capturing this information and how they are able to report the adverse events to the regulatory authorities. Deloitte, through their huge amount of consulting experience, they have come up with Converge Health Safety. Right, IQVIA has been a major player. They are coming up with a vigilance platform. Clinivo already has a one pharmacovigilance software. Logics. So they have another array of software for, and their most prominent one is the PV Signal, right? So this is a very colorful world of PV technology, and like how it is for all you students who are a part of NIPA, you are not only bringing a career on PV, but you can also pursue your career in PV technology. Okay, so there are so many organizations that are looking for young, dynamic people who can move out of their comfort zone and try to be a part of the technology organization. You understand the drugs, you understand the mechanism behind how drugs work, you understand the logistics of drug approval process, right? So why don't you bring that knowledge into technology and try to solve a lot of problems that the pharma will anticipate due to some changing regulatory guidelines or changing disease landscapes and things like that, right? So we need a lot of help and support in the technology space as well. So I would strongly recommend you guys to spend some time understanding, how you can equip yourself with learning some big data analytics, learning, investing your time on understanding what are the different coding languages like Python and all of that, right? And nowadays, like the coding has started from school, like the fifth standard kids are doing coding, right? So what's stopping you from learning these codes? Because these are the future. So tomorrow, when people talk about a certain algorithm, right? Suppose among 10 of you, two of you understand what they are talking about from an algorithm perspective, you automatically outshine the rest of the folks, right? So please consider this as an area where you can invest in terms of along with your um, BPharm, MPharm, PharmD, PhDs, right? Invest in technology, learning technology on how you can bring a change a difference in the way we are doing PV as of today. Second area that I feel is very, very interesting is to learn a foreign language. Okay, to learn a foreign language. We know that with PII coming in force or the GDPR coming in force, the globalization of PV is slowly getting restricted, right? So we know that a certain country person can access certain on the same country's data, right? He may not be able to touch all the country's data, and if he touches, it'll be blinded, not of much use, right? So if you can pick up a language, probably Japanese or Chinese, and these are or Korean, right, or for that matter, German. So these are countries that want people to start doing and processing information in the local language, right? So this is an alternative area that you can parallelly invest your time in learning a new language along with your pharmacy skill. You become a key market leader. You become a very sought after talent, in the market, right? Allows you to do a lot of travel, allows you to see new places, right? You're able to explore how PV is done in different regions of the world, right? So just calling it out here. So we'll take a brief peek into understanding what are the different areas on different safety databases. How do they really look? Okay, essentially all the safety database follow the same process, right? They follow the same process, they follow the same ideology. Okay, they do the same, they fulfill the same purpose, but they become very unique in terms of addressing certain key challenges that are existing in the current market, right? So the reason why we have Argus safety is because one, it is well accepted by the regulatory authorities, has a very comprehensive list. It has evolved over the last 20 years. So the richness of experience that it has, right? It's a huge monolith. It's a very big, the one single platform that brings everything together, right? It's like a magnet. It kind of brings all this information in one single portal, right? And it has evolved over a period of time, and it's always stood up to newer technologies, newer regulatory changes, and things like that. So in Oracle Argus Safety, we have Safety One and we have Argus Safety, right? So these are the two screens, and this is how they look. And we will go through some details around each screen as to what do they really mean and how do we navigate on processing individual case safety report. Then we have the Oracle Safety Empirica signals. So this is a screenshot that I would wanted to show you. That gives you you can see here there are a number of tabs here, right? And here it talks about drug profile. In the drugs, when you look at the drug profile, you can see the kind of adverse events. You can see food poisoning, then you can see pulmonary tuberculosis, food interaction. So it'll give you in the database what are the top 10, 15 adverse events that this particular drug is causing, right? That this particular drug is causing. Then it talks about what all adverse event occurred in what all time duration, right? So you can set your time duration every 2 years, right? What are the key adverse events? Say hepatitis, how it has been performing. So some from red has become green, and from there they become yellow and orange, right? So you can keep a track on that. And this is what I talked about from a heat chart perspective, right? A heat chart, here you can see that there are bigger squares, and then within those bigger squares, there are smaller squares, right? So this big square is your system organ classification, like say cardiovascular system, right? So, and more darker you have these colors, it means that more darker is the problem. More green, more reddish it is, more problematic that area is. So if you're seeing this whole profile for close to 30,000 or maybe 3 lakhs, maybe 300,000 adverse events that are being reported for that particular product, when you spot a graph on a heat chart, it'll tell you that, okay, this cardiovascular system is the most impacted organ system. And in that, if you further zoom in and click on each of these blocks, it'll tell you, is this arrhythmia? Is this cardiomegaly? Is it, heart failure? That is actually the highest among this particular chart. So this sort of charts and analytics is something that it bring value to what we're doing, right? And it's not only in these graphical formats, you also get to see in this tabular format that you can see in the backdrop, right? And then you can generate reports, you can run your own analysis, taking it forward. And then we have RSG, okay, which is called as LifeSphere MI, Medical Information Management. So this tool allows you to capture information about a particular drug for their customers. So this is how it visually looks. Then we have Viva Vault Safety. Okay, so if you can see, there are different tabs around here. and in each tab you have specific information coming in for that particular item, right? So you can see that this has a case so and so is a SUSAR, the event of the urticaria, and the name of the drug. Right? So it kind of gives you a summary around here, and then most of the elements that are shown here, including source document. Right? So you can see source. You can see one PDF that's kind of opening up, and that is on Viva Vault. Clinivo PV database, pretty straight, simple, straightforward. So it talks about case import, duplicate search, open a case, blah, blah, blah. And then you're able to kind of create a ICSR. You have things like what Chusa Polaris. Now, this particular group is very good in terms of extracting information from a scanned document that comes in via email, right? So they will look at a native file, then they will do an OCR, which is optical character recognition, texting. Then they will look at if there any need for translation, and then finally you'll have annotated information. And whatever information is filled here, you'll have it automatically filled into those. So humans don't have to do transcription job, right? Earlier, you would have heard of a concept called as medical transcription. People used to hear audio files and then type it out manually. Nowadays, that market is completely gone, right? Because we have robots who can do that. They're able to listen and then they type. So you have voice to text technology kind of caught up, and then we have no more medical transcriptionist kind of running the shop, right? So here, what we're doing is data entry. Data entry is very manual. It is like copying information from one PDF to inserting it into a field within a safety database. Now that is pretty monotonous and may not require that level of intelligence to basically transcribe that information from one side to another side, right? So we will have to, so we are looking at technologies that can automate a number of activities of this sort. And then we have technologies like Sci Exchange, which basically allows you to submit these different ICSR to regulatory authorities and, like I said, to investigators, to independent ethics committee review board. So where all we would like to have this distributed, we are able to distribute it using some specified technologies and applications. And then we have Vitrana's Highlight, right? So Vitrana's Highlight is also one such tool which works on a completely modular principle, which allows you to be 20% more efficient, has proactive alert system, has an omni-channel experience across. You can even do case processing on your mobile phone, right? You can capture information on the mobile phone and send it to somebody sitting in the central office where you're able to, fill up full data, right? Then we have enhanced language support. So if you're getting information from Portugal, from Brazil, from Argentina, or from France, I think the system is able to auto recognize, and then it uses a lot of these translation APIs to convert them into English and then send them into database. Then it also has different efficiency measures that will allow you to basically move forward effectively. So now we kind of land into the demo. Before we land into a demo, I'll just take a quick pause here. So let me just quickly stop the presentation, move over to my screen, and then we'll kind of come back, okay?
Sir, your mic is muted.
Sorry, I was checking if you can see my screen now.
Yes, we can see. Yeah.
Okay, yeah, thanks. So before we go into the demo, probably we'll keep navigating back and forth to the slide, right? Which basically gives you an idea about what is a case input when we talk about case inputs. So we talk about information coming in from safety call centers, right? So if you have an adverse event, you have a toll-free number that you give them a call and tell them, "Hello, I'm Amar, and I have suffered, severe vomiting because I took your colchicine or chloroquine tablets, and then what should I be doing, right?" So once you give the call center executive will ask you a few questions like, okay, when did you take it? What is the dosage that you took, right? What are you? Did you seek any medical help? Did you have undergo a hospitalization, right? Or what sort of drugs were given to you so that you are free from those symptoms? So all that information that the call center executive asks you, right, will be captured in a certain form, right? We will look at that as one such case input form. Then you have clinical trials. So you know that once you have clinical trials, if you have an SAE, you need to fill up an SAE form, right? But that SAE form is a part of your protocol. So in your protocol, you will have SAE forms attached, or if in your trial master file, you will have SAE forms that will be provided to you by your sponsor, right? Then you have spontaneous reports, which are basically reports that would have kind of come in from either a hospital nurse or myself as a patient, or from a patient relative. If my dad or mom had suffered any adverse event, then I could report this via email or through any other portal for spontaneous reporting. And then finally, literature search. Literature search is when I search for a certain adverse event being reported and published in the literature, right? So I do my searches on PubMed, on Medline, and then try to identify, okay, for my product. So if I'm a GSK and I have a product called Crocin, I'm going to search for Crocin, and all the adverse event, all the literature articles published on Crocin with an adverse event kind of shows up. And then I go through them and then try to understand, okay, is there a attribution, and then create an individual case safety report out of that, right? So these are some of the few case input formats that will kind of come in. So then we need to create a case, right? In case intake, what you do is to create a case. When you say create a case, it means that the case with a unique ID gets formed, right? Once you get that case ID, then it becomes the responsibility of the market authorization holder to submit it, right? So then he would push it into the medical data processing, which will have triage. Triage workflow step, you most would be knowing that their responsibility is to categorize the drugs based on the timelines that they're getting submitted. We know that death cases have to be submitted within 7 days. We know that all serious cases which have, like, hospitalization, congenital anomalies, any other medically significant adverse events, so all of these have to be submitted within 15 days of being made aware. And then if it is non-serious case, things like headache, mild abdominal pain, all of these which are non-serious, right? You can submit it, or you don't have to submit it. If you submit it, you can lock it and keep it in your own case, because the regulatory authorities will not be interested in knowing about your non-serious adverse event. But those non-serious adverse event have to be still database, because your aggregate reports all run on those non-serious reports as well. Then you have data entry, where you're entering almost all the specific details, like how the call center person had asked information about how much of chloroquine did you take, how many times in a day did you take, did the doctor prescribe it to you, did you skip any medications, right? So all that information have to be getting into the database, which is basically done by the data entry person. Then you have a query process. So if you have information that is incomplete, then you query your source, right? It could be a call center. You will contact him, or you will contact the investigator, or or anybody who's reported, or you can even query the author of the article and ask him some specific questions about that. So by doing these two, you're bringing in a good quality. And then finally, you'll have even QC, or quality check, or quality review, which will look at all the essential processes within the system and say, yes, it is good, correct. And if it is required, then you need a medical input, a medical review, right? Once you have the medical reviews inputs, and then you have the causality all defined, then you send it to case closure, right? Case closure results in two things. One, you close the case at this level, right? At the case processing level. There's no more new information that needs to be added, right? Then it is ready. If it is a adverse event that needs to be submitted to regulatory authorities, then it gets into the submission portal, which is a case output submission portal, and gets submitted to the regulatory authorities. And as it gets submitted to regulatory authorities, it sits in the database, from where every 6 months, 9 months, 1 year, you run a report out of that database, and you get an aggregate output. And based on that aggregate output and the individual case report, you will send it to regulatory authorities. Based on these two databases, you will also perform signal analysis. Where we used empirical signal, we use signal analysis, and then we draft the benefit risk assessment and mitigation. So now we will look at the portion that talks about case intake, case processing, and case output. Okay. So what I'm going to do is to quickly look at one such adverse event form. Okay. So we're looking at an adverse event form. I hope you can see. This is an adverse event form, talking about global P adverse event form. So if you are in a call center, right, and you call them, so first thing is they will write down at what date did you call and make them aware. So this is called as a date of awareness. Right. Then they will look at what type of reporting it is. If you had called in a call center, it becomes a spontaneous. If you are a part of the clinical trial or any such protocol driven, you become solicited, right? And then they'll look at all this information. The first time you're reporting, it's called initial, right? If it is a follow-up. Then they will say, who's the person who's reporting, right? What is the patient's name? Which country does this person belong to? Then we look at patient details, right? So we take the initials primarily because we should ensure the confidentiality of the patient should be maintained, right? Then you have the date of birth, then you have female, you have the gender, then you have the height and weight, then you have any other details like vaccination details, past medical history, then relevant diagnostic testing, right? And then what is the suspect drug that the person has taken, right? What is the frequency dose? What is the route that person has taken? Any batch lot number is available for this particular product. So all this information will be captured by the call center person, right? And they will also write a certain narrative that, we got a call from so and so. And the person was also taking aspirin, Zinitac, right? And likewise, they would get it signed and send it to the case processing center. Okay. Now this particular form will be sent via email. What I would do is, as a case processor, we would log into my safety database, which is Argus Safety Database. Okay, so now we'll get into the formal demo of the Argus Safety Database. We have precisely 30 minutes for this discussion, 20 minutes for the demo, and 10 minutes maybe for the questions. I'm assuming that we will have many questions. So one very classical requirement by the regulatory authorities is what is mentioned as compliance to 21 CFR Part 11. Okay, when you talk about 21 CFR Part 11, it means that there has to be a traceability of where the information has come, how the information is done, who has done that information, who has done that particular activity in the PV, and what is the outcome, right? Second is it's also a requirement for the technology providers, primarily because one, they have to keep that signature on to all the activities that I particularly do, but also in their application as to who is using, who is not using it. So it can't be like like a general form that you can go in without a login and full in. So once you log in, you become responsible for anything that is done within the application. Okay, so my administrator would have provided me the username and password to basically log in. Okay, so now I'm logging into the Argus Safety application, and it takes me to this landing page. Okay, so this landing page basically gives me an ability to perform multiple different activities that are required for the PV world. Okay, so let's quickly go through and browse through what are the different features and functionality that are existing in Argus. Just going to zoom in here so that you're able to view this. So Dr. Krishna, just want to double check with you. Is this more visible than earlier?
Yeah, it is much better.
Okay, great. So I might have to zoom out, right? I might have to zoom out, and I have to show you something which requires a bigger picture. But right now I'm zoomed in so that you can see the different components of what's being shown here. Okay. So first it will tell me that I'm logged into Argus Safety, my name as to who is logged in, what is the date and time of login, right? And what is the database. So there will be multiple databases. So if I'm working for a pharma company like say Cipla, right, it will be written Cipla over here. And in Cipla, if you have different databases, like you'll have one database which will be used for production, which means that that's the main database, right? Then they'll have a development database, which means that you can do trial. Then you can have a training database where you can do your trainings, where you can do sample cases and do for trainings, right? And then you'll also have a sandbox environment where technology testings will be done, configuration will be done, and testings will be done, right? So for us, it's important to understand that we are logging into the right database, right? So that is mentioned over here. Then you have home, which will bring you back to here. Then you have help file. If you click on help file, it'll open up and give you information about what each field does and what are some very high level help HTML file will be available for you. Okay, and then you can log out. So once you log out, once you click on log out, you'll see that the system will ask you to, it will close all the open cases and it'll bring you back here, right? So let me log in back. Let me log back in. So I log back in and it brings me back to the same screen, right? And you can see that whatever I had zoomed remains zoomed, it didn't go back. So that's good news. Then you have the first list of tabs, what we call it as. So the most important one is your case action tab, right? It allows you to open an existing case or create a new case. And third is create a new case from image that's already available, right? Then you have here active cases. It'll tell me what is the last case that I worked on, right? Then you have work list, which says what is my list of tasks that I have pending for me, right? So if I click on new work list, it'll tell me what are the new cases that are available. If I want to have open, tell me what case, action items. Action items are some tasks that are assigned to you, so you can get to see how many action items are being assigned. So you have multiple such functionality to basically view. So let's quickly click on action items tab here. So you can see that these are the cases that has action items. So this is how it will be shown that, okay, you're in work list, you're in action items. You can search for a certain case. You can type the name of the case and it'll be shown up, right? Or you can see from the entire list. It'll give you a snapshot or a summary to say what type of case is it. It is a serious case. It says J amended description. It's sent to Japan. It was opened on the 8th of December. Due date has not been mentioned. 1,000 days that it has been open. It has not been assigned to anyone and not been completed. So these sort of lists are what is the part of work list. So we talked about case action, you're opening new work list, right? Then you have something called reports. Reports are, if you want to do a compliance report, it'll give you to which regulatory authorities you were compliant. Out of 100 cases that you have completed, how many did you complete on time? How many got submitted on time, right? So that kind of report. Aggregate report allows you to create case data analysis and multiple different listings that can be put into the aggregate reports, right? So, and this periodic report allows you to create a clinical trial periodic report, a PSUR, a periodic safety update report, an IND and NDA. And likewise, the different reporting. Then you also have a local affiliate, which is like if you're a part of the local country, then what kind of work you can do. Then in the utilities, you have meta drug user, then you have audit logs, error logs, you can perform undeletion of a case, right? You can do multiple different activities of that sort here. Then you have dashboards. Dashboards will give you an idea about how many cases are open, how many action items are open, how many did you close, right? What sort of signals are available? What is the expedited report status? How many are submitted? How many are pending? How many are waiting to be submitted? Right. So it'll kind of give you all of that. And then you have an Argus console. Argus console is the heart of the Argus. Okay, so this basically has all the settings that has been based on which this whole application will kind of run. Okay, this is a whole chapter on its own. Like say, for example, if I click on that, it kind of opens up a list of items over here where we talk about code list. Like say, for example, this is where all your settings are there. Like say for right now, it will tell you what are the countries that you will get in your drop downs, right? So you're talking about, close to how many countries? 251 countries, right? So all of them are set up over here. What is the A2 code? What is the A3 code? What is the numeric code? So these are all international standards based on which we have all this information sitting in the background, right? So knowing Argus code console is the role of an administrator. Okay, as a user, somebody who's entering the information into Argus Safety will not have access to it. This is more like an administrator. So tomorrow you want to add a new country, right? So here is the person who will say add a new country, and then you can add all this information, and it will be available for the case processor to process the case. Okay. So likewise, there are multiple different items like things like, gender, things like, message type, occupation. So you have male, female, other unknown, right? So you have all of these kind of stored in here. Not only that, it also has a list of all the products and their related licenses. How do you want to configure? Because every product will have a product family, right? Then it will have the ingredients marked as a part of the family, and then it will also have a certain license associated with that, right? License, when we say license, things like, it is an investigational license, which is your IND, and then it'll have a post-marketing license or NDA, which is a new drug application. And if it's an ANDA, right, then it will have an ANDA license. Why these are important? It is important because based on the license is when the organization will categorize the case to be submitted to the regulatory authorities. What type of PSURs need to be submitted as a part of the aggregate reports, right? So all of this basically are lined up here in products and licenses. In fact, there are many different, and if you're configuring a clinical trial, right, so it'll go into a studies configuration. So you can click on the studies and you're able to enter a new clinical trial, right? So let's look at this study called as Paravitrana. So this is the product. Okay, and it will have a clinical trial with a study ID, project ID, and all of that, right, allows you to understand that, okay, so we have a source that is coming in from a clinical trial. Okay. Then we have the access management, which will have adding a new user, right, adding a new group, adding a new clinical trial site, all of this kind of comes in. Then you have system configurations, like how do you determine the case priority? What are the different field labels that you have? So you see study name is one such label, right? So if you want to add a new label, right, you can add them. You can also define the workflow to say from case intake to data entry to medical review to quality review and then to submissions. It will have a certain predefined workflow that has been set. Okay, so these are certain key features of what you call as a part of your console screen. So administrator, if you be a part of these technology companies that I showed you, right? Then for you, it's important to understand how Argus works based on the Argus console. What we'll do quickly is to kind of create a case, right? And show you how in Argus it will help us to create a case in the application. Okay. So what I'm going to do is I'm going to create a new case. Then I have the screen that's kind of come up. Okay, so if you look at this, it says, okay, there's a initial receipt date. So if you say initial receipt date is the date when first time the patient initially gives me that information, right? So if I use this as 20th September 2021, right? And then central receipt date will miss that. And then we have country as United States. So it'll pick up the country as United States. Right, and then you have the report type, right? So you can take the report type. You have the report type as clinical trial, right? So you have clinical trial here, or for that matter, let's use it as spontaneous report, right? Because we're looking at spontaneous reports. Then you have, say, the product name. Which product did the patient, which product is he reporting the side effect for, right? Say, for example, we are looking at Rivalusimab. So it allows you to select the product name. Right? So you can select the product in United States. It's going to search for all products which are available within United States, right? So you can select the product. Let me see which products get selected. Okay, none of them are getting selected. Let's look at changing this to. Okay. So description as reported. So I'm going to talk about description as reported to be, say, vomiting, right? Once you click on vomiting, right, it would say across here. So you click on that, it will open up the Medra dictionary and it will start coding that, right? So if you see here, some of them are marked in orange, right? All the ones that are marked in orange are basically mandatory fields. It will also ask vomiting, vomiting, when is the onset date? When is the onset date, right? So say, for example, if this was on 18th September, it will be onset date. And who's the reporter, right? So if it could be a doctor, so-and-so, right? I'm going to put my name. And we have the patient first name or last name. If we have, you know, patient ID, date of birth of the patient. And then I say search. If I say search, what it does is it will go back to my existing information in my database, and it will say that, oh, this person is already available. So here it's saying no case found, right? So if it's already available, then we don't have to create a new case, right? You can just add it as a follow-up. So right now we don't have any cases existing in the system. We're saying just continue. Now it is asking, okay, what is the seriousness criteria? Okay, seriousness criteria, I'm going to choose it as medically significant. And then anything, the causality has been reported by the patient, unknown, right? So once I do this, and then I click on attachments and references. So I'm going to add this attachment. So I'm going to click on classification. I'm going to say that this is a little, this is not a little. This could be, source document, and I'm going to say file attachment. So it's asking me to open the attachment here. So I browse. I have the SAE form. I'm going to attach that. Oh, it's already in use. Let me close that. Now you can see that it gets getting attached to the case. Okay, you can see that there's an SAE form, file attachment. Okay. Now I click on book in. So once I click on book in, what happens is the system will open up a new case with the full case details, right? And it'll assign me a case number. 21 IN 001571 has been successfully created. You wish to open the case, right? Now, if I'm a person who just does this intake work, right, I'll say no and I'll start creating another case, right? And this case that has been created will be sitting in the case processing or data entry step. Okay, so I click no and then I start doing the next case. I'll keep booking in. So this step is called as the intake step or the book-in step. Okay, so just imagine that, okay, I've finished the book-in step and now I am a data entry person. So I click on that. Now this is my case form for this particular case. Okay, for data entry. Now, if I go to activities, I will find that there are additional information. You'll find that there is a source document that is attached here, right? So I click on that. My source document gets opened up here, right? So while that is getting opened up, let's go back and look at what are the different information that we need to write. So we have this entire case form distributed as general information, patient information, products, events, some analysis, any activities where we talked about assigning some tasks to some people, right? So that one, right? Activities and any additional information that you'd like to add and understand what are the regulatory reports that will get submitted out of it, right? So now we entered that basic medical information so that we're able to create this case. Now in this, we need to add more information, right? Which are there here. So we're looking at, okay, we change this to 19 September. So go back. Say, oh, okay, so this is sorry. Okay, so let me align it to the source document. It says solicited as a spontaneous. Okay, I can see here report type spontaneous. We change it to solicited. Yeah, solicited case. Country of origin is India. Then local case reference ID. Where should we put the reference ID? The reference ID should be somewhere in additional information. You see references here, right? So we click on the type, the ID here. Okay, put that as local case. Okay. We kind of put that information there. Initial or follow-up. Where is initial or follow-up? Go to general. We have it as initial receipt date, initial justification. So this is an initial case, right? So there are no follow-up information there. Then you have healthcare professional, correspondence inbound, outbound. Yes, it's man v man, patient or consumer. We don't have information. Patient initial decay 2, 1, 2, 3, 2. Let's go back and see on patients tab. You have patient sponsor identifier patient ID, right? So we put that over here, patient ID. Okay, he's an adult. Date of birth is 6/6/1987. Let's correct the date of birth that we put in. 6/6/1987. 34 years. Age is female. Gender is female. Right? Then once you click on female, pregnancy gets enabled, right? So you will keep height 154, height is 154 centimeters. Okay, weight is 54 kgs. Weight is 54 kgs. It gets automatically selected. Allows you to change to pounds. Here it's 72 inches. Okay. Then you have vaccination at age, vaccination units, vaccination name, vaccination date, age at vaccination. Okay, we're just going to ignore it and come back later on. Diagnostic types, it has hemoglobin, result 12.5. So where do we find here? Lab data. So we are saying add the test. Hemoglobin. Okay, once you say hemoglobin, okay, it gets coded. You see Meta browser opening up, it gets automatically coded and it says the checkbox is done. Here we'll add a date. The date here that it is showing as 5/5/2020, 05/05/20. Okay. Here what we do is say add value is 12.5. 12.5. What is the units? Percentage. Oops. 12.5. This is normal. Okay. So if you have more, you keep adding more, like on different dates, like say 10th May 2020. Keep adding another one. So this was 14.5. This is normal. Okay. Here it is percentage. So automatically that gets picked up over there. And then you have the lower range of normal, 12 to 16. Okay. So you added the hemoglobin information. Then you talk about the product. It says Bicoseuls. Okay. Then you go to products. Then we have used Crocin, right? Bicoseuls. Okay. You see Bicoseuls is here. So I select it. Gets coded. It talks about what the generic name. The indication, do we have the indication, dose frequency, initial dose 500 mg, IV, lot number, batch number. So we have all that information here, right? Dosage regimen, start date. We have the start date, yeah, you have the start date ongoing, right? So it is 17 February 2020 is ongoing, right? It gets grayed out. Dose is 500 mg. Frequency is once. You actually have a drop down here where you can choose QOD, okay, daily dosage, milligram, units, route of administration, I say intravenous, current route of administration, accidental expert. So you're able to fill up all the all the drug related indication as vitamin deficiency, right? So kind of missed that over here. Reported indication is vitamin deficiency. Yeah, again, this, okay, so I've written a wrong spelling, so it's stopped over there, right? Deficiency, right? Okay, so you have vitamin deficiency. So vitaminosis, vitamin related, someone is say select. So you see that's checked over here. You see tick box kind of coming in here, right? So you end up, what is the adverse event? You have hair fall, vomiting, accident, hypertension, right? You go to the event section and then we already written vomiting. Okay, so we look at vomiting, 10th phase one. So no fatal, nothing unknown. Okay, so here you have other details that you need to kind of see if you want to write. So here it says it's not coded, so you can click on your screen to see if you can code this. Now remove one T. just coded vomiting, 2 T's, remove 1 T, and there you go, right? So you've entered the event details, right? In the event details, you have something called as event assessment. Okay, so here you choose the product and you choose the product causality. Okay, then you have the data sheets. Data sheets are company core data sheets and the other data sheets. And then you have whatever license that are assigned, right? And then um, it will recalculate and provide you the information. And in the product event details, you'll mention the product and the event and whatever causality that kind of comes up, right? So if you go to the event, there are also other details like you can mark lack of efficacy, progression of disease, drug withdrawal, is it dropped from the study due to an event, and so on and so forth, right? You can also add nature of the event, right? What is accidental, right? Or something. So this is a drop down. So these are precise information that are kind of coming in, right? You go to products, there are some more additional information right at the bottom, which talks about action taken, right? So did you no change in the action taken? De-challenge results, right? So if you say this is stopped, right? If you say dosage, no changes, okay? If I choose withdrawn, right? Once you say withdrawn, then your de-challenge results say positive. And on what date was it positive, right? So you need to mention that bit as well. Once you finish de-challenge, say no, re-challenge, is it positive or negative? You say negative. So these, if they are reported by the patient, it becomes helpful for the medical reviewers. Okay. So if it's a device, then you need to enter device related information. If it's a device, okay. So what sort of device it is, all sort of device information needs to be filled in, right? If it's a vaccine, right, then all vaccine related information needs to go into the system, which is like what is the start date, small date, or route of administration, all of that stuff. Once we do the information, once we have filled up the information events, so you can keep adding as many events as you want. Second thing that you can see here is say suspect, right? You can also say it's a symptom. It's not suspect, it is a symptom, right? So um, you can talk about, like, say, for example, the actual diagnosis, right? So if the diagnosis is intussusception, right? Intussusception. Okay, let's make it simple, like appendicitis. Okay, appendicitis will be the diagnosis, okay? Says diagnosis or symptom, right? So if I say diagnosis, it remains a diagnosis. Let me make this appendicitis. Now we are able to create a case for appendicitis, hospitalized. And then it tells you when did you start hospitalization? 20th May 2020. And then event cost hospitalization, say, okay, you have the details. So you see here, and what you could do is you can reorder them, right? So, and then you can also click relationship where you're bringing appendicitis up. So all symptoms can be brought under a certain diagnosis. Okay, now this is symptom, right? So I'm going to make this symptom and I'm going to make this as diagnosis. You see on H here, H basically says that the person, this led to hospitalization. Now you can see that vomiting can be a part of appendicitis. Because of appendicitis, he had vomiting. So why is this important? In your event assessment screen, your events, which is right now not showing up, will basically say that you don't have to do any sort of an assessment for vomiting. Appendicitis assessment is good enough. So quick time check. We just a little ahead of our time. So you also can write a narrative if you can. If you go in here and then there is a narrative written here, you can either copy and paste it, right? And or you can generate a narrative. What this does is it will go into the system and capture all the information and then put it into the system, like say append. So you'll see that there'll be a new line of information that has come in, right? So this information was not there and now that has been added on to that. So here you also mentioned case serious, causality, yes, listedness, determination, and so on and so forth, right? So you're able to do that. Then you have activities which allows you to capture any sort of letters that you want to send to a reporter, right? Or anything of that sort. All that kind of comes in. And then what you do is there is this button called as forward case, right? So once you click on forward case, it will allow you to move it to the next workflow step. So from entry, you can move it. So you can see right now it says data entry, right? If you click forward, you could also say that, it has to go into quality review. And you can route it to any specific user who's a part of your team, right? Or you just leave it at any, the manager will allocate this case. I'm going to write case data entry complete. So it's kind of routed to, okay, so it'll be saved and closed. So now what happens is that case has been saved successfully and you're back into normal. So your job as a data entry person gets over, right? Let's go back and see what happened to this particular case. Where is it sitting? Now it's sitting in quality review, you see, right? And then you can also forward this to medical review, right? So you can send it to medical review. Did. Okay, I'm going to send it to medical review. It has your, okay, restrictions. Case has been sent to medical review. Now let's look at where is this particular case. This case right now will be in medical review, right? In medical review, there is a specific screen for medical review, right? So this is the medical review screen. You click on the medical review screen and all the information that has been written in that particular case will be visible. And then the medical reviewers can enter the comments, right? Vomiting is related to the product Decosiles, right? So I'm going to make this sort of case comment, company comment, and I'm going to say, okay, it's a serious agent is causal listed, it is listed, okay. Mostly these are all auto-calculated values, okay. And I'm just going to make sure that all this information gets populated. Then there is a temporal view, which says when the drug was taken, when the appendicitis came up, what time vomiting happened, when the patient was hospitalized. It kind of gives you a timeline summary of where we are, allows the medical reviewer to basically arrive at a certain time-related conclusion. Okay, then he can even assign action items. So once this is done, right, I can forward the case. I can forward the case to so-called submission. Okay, submission is the last and final step, right? Medical review completed. If you see here, you would have seen that, okay, it came into initial case, data entry, I routed it. quality review, routed it, medical review, it's sent from quality review to medical review. So you'll get the history of all that happened to this particular case, right? Then it's click it to submission, and then it's gone into submission workflow step. From here onwards, if there is a requirement that it has to be submitted to the regulatory authorities, it will get automatically submitted to the regulatory authorities. So here, report generation and submission, right? So I leave it to any. Okay, case ready for submission. I say okay, I send it. So now you have the case that is sent and ready to be. So let's take an example of what happens to one such case which has some regulatory reports that are generated. So in the last tab of regulatory report tab, you'll see that there are 3 reports that have been generated. 3 are expirated reports. They are pending by destination. It'll say, oh, it has been sent to FDA. And these are the 2 items. One is marked as non-submit by destination. This is the one that is marked as non-submit by destination. Right? You can click on final. When you click on final, you'll get to see the actual report that is submitted to the regulatory authorities. Okay. Then you can say schedule new report or auto schedule. When you say auto schedule, it will schedule a report and keep it ready for submission to the regulatory authorities. Okay, so this session generally goes on for like probably a month of training where you get to use all the different features, functionalities, like say, for example, it allows you to copy a case, save a case, close, do medical review, coding review, all of this stuff, right? So I think we have this limited time frame. We've been kind of extending and I'm eating into your lunch times. So I'll just take a pause right now and quickly flip back to my PowerPoint. And the last slide, which talks about the future, right? So we need to be technologically ready for regulatory updates, right? That's a common thing that will kind of come in. Best in class data quality, right? How do we manage combination products? How can we come up with real-time data analytics? Natural language processing and signal detections, translations and local language support, patient identity identifiers, global data protection regulations. How can we remain compliant? Automations and machine learning, right? How do we look at connected systems? What we talked about, device variables, what we talked about, one database talking to the other database, and then standardizations of some of the existing reports, right? And how do we have an integrated data across clinical safety and real world evidence, right? So these are some of the areas that we are constantly working on from a technology perspective and making sure that the technology is catching up and is accommodating these new needs and requirements that are available in the market. Okay. So team, thank you so much for your patient hearing. I'll be more than welcome to take your questions and answer your queries at this hour.
Thank you, sir. Thank you very much for explaining the Argus and all other various aspects of databases, what commonly using in pharmacovigilance. Actually, this was a dream for me and I think many of us to see at least Argus, how exactly it looks like. Because they could have seen many other databases in terms of academics and PvPI, which if and all. But I searched many times, know who will be the best person to explain the Argus, and I rightly got you. Thank you so much for connecting with us and explaining all these concepts. I'm sure that at least participants are enjoyed by seeing this Argus, how exactly it looks like. Maybe when they go to industry, at least they will have some basic idea how exactly the Argus works, so that they can maybe develop for the once they go to industry. Thank you very much for all that support and explanation. And I'm seeing the messages. Students are enjoying your session, at least by seeing that Argus and the explanation what you are given. The very easy way, I think they understood all the concepts. I think one question I can see there, what are the causality assessments are available in the Argus?
See, the beauty is every organization have their own way of organizing their causality or relatedness, right? Argus is just a tool. It's just a house where you can go and stay. So where you're asking, where should I put the shelf? The house is yours. You can put the shelf wherever you want. So depending upon the organization's policies and SOPs and procedures, they can configure what sort of causality that Argus should show to the user, right? It could be the Naranjo system, it could be a 5-scale rating system, or it could be just a binary. It's related, not related, right? Most organizations are going that route where we are not really looking at probable, possible, all of that is getting wiped out. And they said, we don't want these gray areas. Let's look at only binary. It's related or not related, right? So these sort of causalities. What technology provides is to give that free hand to the organization to use the technology as a tool, right? And the technology should not kind of become a restrictive factor, right? So it should not have something which is very rigid. It should be rigid, but at the same time flexible in terms of allowing people to configure a lot of things by their own.
Exactly. And I think by this your lecture, what we understood is Argus is a software. It is not meant for only for one function in pharmacovigilance. I think we can do multiple things by using Argus in industry. I think that's what I understand, at least. Plus, from the case processing till signal detection, I think most of the things will happen through Argus. I think so in the industry. Yeah. Thank you very much, sir. Once again, from NIPER side, thank you for connecting with us and explaining all the concepts. Students are enjoyed thoroughly. Thank you. And we will be in touch with you for future. If you require anything related to pharmacovigilance, we will be in touch with you certainly.
Absolutely. My pleasure. And I would encourage your students of NIPER who are very clever, intelligent. When we talked about learning a few coding skills, right? Learning some of the language skills, right? So we'll really, really put you in a very strong spot, right? Also, it creates the differentiation factor.
Yes.
It's very competitive world, right? Third is more importantly, machines are taking over a lot of our roles, right? We need to start looking at what we are really good at, which is more like, the intelligence, the emotional quotient, how are we able to manage multitask, right? So those are the things that machines will take a lot of time to catch up. And that's where our true value and worth is. And therefore, wishing you all the best and all your students best of luck for their future endeavors. Thank you so much for this opportunity.
Thank you. Thank you very much once again. So the participants, so with this, we come to the end of scientific sessions. Thank you for being with us all the 2 days. And it is my responsibility to thank all the people who involved directly or indirectly for the successful workshop on pharmacovigilance, starting from Chief Guest of our function, Sri Bhagwant Kubaji, the Honorable Minister for State for Chemicals and Fertilizers, Government of India. And then the keynote address was delivered by Professor Vaika Gupta sir, who was the president of AIMS Bhopal and AIMS Jammu. And the guest of honors were Mr. Sten Olsen, who joined from Sweden. He was a past president for ISOP. And another guest of honor, we had Dr. Jay Prakash. He was a senior principal scientific officer at the Indian Pharmacopy Commission, doing wonderful job for the Pharmacovigilance Program of India. And followed by, we have various speakers, eminent speakers. I'm sure that all of you enjoyed the sessions by the eminent speakers who joined from both industry and academia as well, especially like Dr. Puneet Damija, Dr. Ananya Chakraborty. She explained very nicely about the VG-flow, demonstrating very clearly about the VG-flow, how we need to use it. And the review of quality of ADS, it was very well explained by Dr. Harmeet Singh Rehan sir. And followed by, we had a signal detection session by Dr. Varun Vaidya. And even I have explained few things about the signal detection, especially how exactly the VG lice looks like and what kind of analysis we are going to be doing the VG lice. So that was we had yesterday. And today morning, it was started very well. I'm seeing the messages, all are enjoying the sessions from the morning. Subish explained about data mining in public drug safety databases by demonstrating the open vigil, which is very useful for doing any kind of research by taking the openly available data. And we can come out with certainly a good number of publications. And also, we have seen the experience talk from Dr. Moindon, who was a president and CEO of PVCon Consulting Services. And he brought all his experience in terms of pharmacovigilance and explained all the concepts very well. And finally, I think we had an opportunity to see at least Argus, how exactly it looks like and how it is meant for and for what purpose the industries are using this Argus. I think it is very clear for all of you. by this time, and it was explained by Dr. Amarjit Raja. Thank you so much. So I thank each and every one, all the speakers and delegates, and my special thanks to my team of people who are working for this workshop, especially the technical team, Sumon and Abhijit. They're there with me from yesterday. Thank you, everyone. Thank you to all the participants for joining with us, and I hope you enjoyed the 2 day sessions. And all the credits once again goes to Naipar Guwahati, who extended their support for conducting this workshop. And most importantly, I need to thank IPC, Indian Pharmacopoeia Commission, the Pharmacovigilance Program of India, for providing funding for organizing this particular workshop, the 2 days workshop. Thanks to each and every one. I'm sure that I think all of you are waiting for this feedback link so that you can fill the form and get the certificate. So my suggestion, personal suggestion, the last one for the day is whenever you find any brochures for these kind of conferences, don't jump in and try to join for the conferences for the purpose of or for the sake of certificate. Okay. So maybe you can join if you feel that the sessions are very interesting and the speakers are very interesting. Maybe you can join and gain the knowledge. So along with the knowledge, if you have certificate, that will be well and good. If you have only certificate but you don't have any knowledge, tomorrow when you go for interview and when you show the certificate, and if anyone asks you any question related to that workshop, if you cannot able to explain, so there is no use of having that certificate, right? So it's always better to have the certificate, but more than that, to have the knowledge, understanding is very, very important. Okay. So finally, I'm posting here the feedback form. You can quickly fill that form, and whoever is filling the form, you will receive the certificate soon. Soon in the sense, at least wait for 3-4 days, because we need to generate the certificates. And please don't message me before 3-4 days at least that you did not receive the certificate. So fill this form, feedback form, and so that we can try to improve more when we come back to you with more and more workshops. Thank you so much, everyone. Please share.